Good morning, everyone. I'm Dr. Tamanna Nasli, Research Officer from CCRU. I welcome you all in the morning session 1 on Unani medicine for globalized health unlocking new opportunities. We have a fascinating lineup of presentations today and a renowned expert. Can we have a floral welcome? Professor Rajagopal, a renowned expert in pharmaceutical sciences, specializing in natural product medicine for women health. With over 71 international publications, 2 patents, and 15 international awards, Dr. Mogana is a distinguished academic and researcher. Currently, she serves as a professor and dean of the Faculty of Pharmaceutical Sciences at UCSI University, Malaysia. Please join me in welcoming Dr. Mogana Sundari Rajagopal. I request Dr. Musti Hassan to give a floral welcome to Madam. Now, may I call upon Professor Arzina Jabin, Assistant Director at National Research Institute of Unani Medicine for Skin Disorders, Hyderabad. She is a distinguished Unani researcher. She has a strong background in general medicine with a gold medal from Dr. NTR University. Professor Jabi has published several research papers and guided many MD and PhD scholars. We have honor to have Dr. Surinder Singh, a stalwart in his field, needs no introduction, with over 100 research papers and 6 books and 48 guided theses. His academic credentials are impeccable. He has completed 20 research projects and received numerous awards, including the ICMR Award for Biomedical Research, AIMS Excellence Research Award, and Dr. B. Mukherjee Prize. His contributions to the fields are truly remarkable. May I call Dr. Mantisha to please give a floral welcome to Dr. Surinder Singh. Please be seated. So, without further delay, let's get started. Uh, uh, Sir, ready?
You ready?
So, I request Surinder sir to call our first speaker of today.
Very good morning. Uh, in a first day of Unani Day, means first scientific day, I'll call first speaker is Dr. Mohammad Idris, the senior principal scientist at CCMB CSIR lab in Hyderabad. With over a 25-year experience in development biology, molecular diagnostic, DNA and proteomics analysis, Dr. Idris has a pioneer groundbreaking study which has identified hundreds of genes, protein, signaling pathway involving in regeneration, and CCMB is famous for this, for finding hundreds and thousands of genes. His main research has light on a critical aspect of a cell cycle regulation, nervous system development, cellular growth, and aging on regenerative disease. He has contributed significantly on an Indian Pharmacopoeia Commission monograph on herbal drug through DNA barcode analysis, and it is the only pharmacopoeia among the indigenous system which have a DNA barcode analysis, and the contribution of CCMB and in a very particular Dr. Mohammad Idris is a significant one. He has collaborated with very leading laboratories in India, the sister concern of CSIR labs, Italy, Sweden and Saudi Arabia, and we hope that his scientific contribution continue to make a significant contribution in the field of biomedical sciences and regenerative medicine. Today he will be talking about the DNA barcode library in authenticated plant, the base drug in a Unani pharmacopoeia and a medicine. Dr. Idris. Please.
Yes. Assalamu alaikum. Good morning and namaskar. At the outset, I'd like to thank organizers for giving me this opportunity. So, before presenting, I'd like to thank all the people who are here and going to listen to my talk. So, my talk is about generation of DNA barcode library for authenticating plant-based drug in Unani pharmacopoeia. So, for me, plant doesn't matter or animal doesn't matter. It's all the 4 codons, A, G, T, C. You give me any sample, either it's a plant or animal tissue or fungus or bacteria. We do with the DNA barcoding. We identify them based on their barcode, the DNA sequence. So DNA barcoding is a molecular technique used for species identification. It can be anything based on the unique DNA sequence from specific genomic regions. So you know that any living organism made of DNA as the genetic material. So we go to the specific region of the DNA and sequence them to make it a signature-based identification. So this method enables rapid and accurate classification of organism across diverse taxa, making it a powerful tool in research like biodiversity, conservation, forensic analysis, and identification of novel plants. So DNA barcode is nothing but a short and it's a very short sequence, standardized, typically 200 to 800 base pair long, which is universal within a group. The same genetic region is used for barcoding within a taxonomic group, and it is species-specific variation is highly conserved within a species but variable between species. So we can identify any species within species and genus based on this sequence. Okay. So what are the common barcode regions being used for across the animal kingdom? So for animals like human being or any vertebrates, it's mostly the COI gene, cytochrome C oxidase 1 gene, which is a mitochondrial genome. So you should know that in a cell we have nucleus, mitochondria, but in a plant cell we have also a chloroplast. So we generally extract the DNA from these DNA, the nucleus and the mitochondria in chloroplast, also the chloroplast in plant. So for animal, it is identification based on mitochondrial, which is a high mutation rate, good species resolution is possible. Whereas in plant, we have nuclear and chloroplast genome identification. We don't use mitochondria because they show the DNA evolves too slowly in them. So these are the major genes which have been used to identify the plant species for DNA barcoding. And also fungi and bacteria, similarly, we can identify from their nuclear and ribosomal DNA. So the common used genes for DNA barcoding in plants are ITS1 and 2, RBCL, MATK gene, PSBA gene, and tRNK and tRNL gene. So mostly people are using ITS1 and RBCL, but to identify subspecies, species-specific identification, we have to get into the other genes. So unlike animals, the mitochondrial COA oxidase gene is widely used for barcoding, but plant mitochondrial DNA evolves slowly. So we have to use both nuclear and chloroplast nuclear gene regions as commonly used for accurate species identification. So the most, as I told, it is ITS1 and 2 are widely used, and the application of plant DNA barcoding is, as I told you, it is species identification, helps in identifying known or unknown cryptic plant species, biodiversity conservation, like track species in ecosystem, and detect invasive species. Like this is a common problem in our country. This is a big problem in our country, so we can use this barcoding method to identify the invasive species, and also helping in cataloging plant diversity and detecting endangered species. This is also we are now doing with the Unani system. And also it is useful for authentication the medicinal plants, ensures correct identification of plants in herbal medicine and reducing adulteration risk. and also it's useful for ecological and forensic botany. There are a few limitations in DNA barcoding because of it's not complete database is available, low resolution in certain group, which is still evolving in a big way. Soon this limitation will be addressed. Now coming to why we are using this DNA barcode. So here I'm representing a Ashoka plant. Here you see this is the Ashoka sarcasa, the real original Ashoka, which has a big medicinal value. Whereas this is the duplicate Ashoka, which is nothing but Polyethalia longifolia. We see in the tree, in the roads commonly the long tree, Ashoka tree. But this bark doesn't have any medicinal plants. See, so then we can use the DNA barcode here as a genetic barcode, distinguish one species from another, even if the species looks similar to other or in a different life stage. So here I can extract the DNA from a root, leaf, bark, anything. Any tissue you give, I can extract. So this is the two bark of Sarcasa, Ashoka, and the duplicate Ashoka. So you can by any visual identification, it's very difficult, or botanical identification is so difficult. So how we do is we collect the sample. It can be root, leaf, stem, flower, seed, or any powder form of the plant materials. We'll extract the DNA, we'll amplify using the specific primers and sequence them and analyze them. So how it works? So this is simple like DNA preparation, extracting genomic DNA, amplification, sequencing, and analysis. So this is a cell, how it looks. If I go into any cell, it's a leaf cell, where the green is the chloroplast. So you can see the nucleus, the chloroplast, and the mitochondria. So we can extract the DNA from all these three components from the plant cell, and we can use these following set of primers we use. Nearly 16 genes we are using, because we are not doing monocoding. We are using multi-target coding for like following genes being used to identify the plants. So the ITS1 and 2 genes are from nucleus, and remaining all are from chloroplast. This is a chloroplast, such a big 150 kilobase per gene size, nearly. So this all started when we had a project from Indian Pharmacopoeia Commission. So they want to update the monograph with the DNA barcode for their herbal plants, which they have a monograph in their Indian Pharmacopoeia Commission. So we did, like they gave this powdered form of some plant material. We don't know what is this. So we extracted the DNA, we did the PCR, this is how the PCR looks, and we identified the species as nothing but our neem. Similarly, we developed the barcode. Here we see given 5 barcode of different gene region. So this is all now amended in the monograph of Indian Pharmacopoeia Commission. And they give another sample, which is nothing but a resin, but it is contaminated with other materials of plant. So we simply refuse that. We cannot give the sequence, it is contaminated. And now here you can see Eclipta alba, we gave the barcode, and Gymnicera sylvestre, we gave the barcode, and also we gave Withania somnifera. A few examples I'm showing. So here you see the powder we got Withania somnifera, which we don't know. We amplified, purified, see the different target of different size. Then we sequence them. We got the barcode. See, this is for ITS1, RBCL, and other gene like PSBA and TRNL gene. So this is all like genes which are not there in the other system like UK Pharmacopoeia or US Pharmacopoeia. But we have amended with multi-barcode as a signature. And this is for Amaranthus tricolor. We gave this the first, I think, sequence I got from our Indian Amaranthus species. So this is how the monograph been amended, and we gave the report. So based on this, when we were discussing with Unani system and Unani like NRI UMSD, so we developed a collaborative project with CCMB and CCRM NRI UMSD at Hyderabad for development of DNA barcode library of plant origin single regs of Unani Pharmacopoeia for authentication of plant species used in Unani system, because they were never done any barcode in Unani system. So ours is the first study in this way to approach the plant. So the most common traditional authentication of Unani single drugs includes botanical identification such as taxonomy, macroscopy, and macroscopic examination. And also they use advanced chemical methods, but these techniques have need a whole plant, and it is difficult to apply to forms where plant morphology cannot be determined, and each has certain limitations. So among all the recently developed method, DNA is the most well established, as I told you in the beginning of my talk, and DNA barcoding is now officially recognized as a method for authentication of drug. So we use this method for identifying local documentation of nearly 338 medicinal plants, which have been officially used for Unani system as a single drug. So use of correct plant species is a basic requirement to get the desired benefits of Unani. So with this approach, we started doing this work. So DNA barcodes and reference to authenticate the plant origin of single drug will be done for development of Unani Pharmacopoeia of India. So what is that we do is we are generating the DNA barcode for Unani medicinal plants to create reference DNA library and to authenticate UP controversial Unani Pharmacopoeia controversial drugs from markets using UPA or DBL. And the significance are submission of single plant drug will be with the DNA barcode sequence in international recognized database will be there, and technique can be utilized for effective authentication of the drugs, and update of Unani Pharmacopoeia of India will be there. So these are the different few plants we selected. You can see you must, if Unani based system people are here, they can identify what are these. So we have selected some to start with some 65 plants, and we have generated whatever plant tissue we get. I don't care with those tissue or plant. We just generate the sequence like this and do the analysis. So this all started with Kashif. Dr. Kashif is here. So when he came to lab, he got me 5 badams in powdered form. I don't know what is that. I know badam, which we eat generally, but he told there are 5 badams are here: Delhi badam, Mumbai badam, UP badam, and Kashmir badam, and one Hyderabad badam, which is bitter. So when we said, okay, let's see what is there in the barcode. So when we started the work and we performed the sequence analysis and did the cluster hierarchy, we got all the genes are clustered together, but this Hyderabad badam is separated in the cluster. So unfortunately, I had to haze this sequence because it is live and I don't want to show the sequence. This is unpublished data. So when we looked at the ITS1 and 2 gene, we didn't find any difference. Means there is no change in the sequence for all the 5 badams. So they all are similar at ITS1, 2 gene, and also in RBCL, which has been generally used as a signature barcode target or the signature. But when we went to the next gene, PSVA, we found a big change in the sequence here. You can see here polystitch of one of the nucleotide, whereas this is not there in the other badam. This is the badam of Delhi, which is sweet, and this is the badam of Hyderabad, which is bitter, but has a medicinal value, if I'm not wrong. So we found a big change in the sequence. So here we got the target. So if I get the badam, any preparation, if I go to directly to this PSBA gene as a signature, I can easily identify whether it is adulterated or not, or it is original species or not. And also we got a big base pair deletion in the badam of Hyderabad. That makes it unique for Hyderabad badam. So similarly, so now we are kind of our botanist and scientist at NRMS are confused whether it is Hyenocarpus or Prunus or it is the other one, Hyanensis. So we are now still working on with other genes to place it in the right species classification, which will be done soon and it will be properly classified. Then it came the story of our Pushpa. Pushpa is nothing but the sandal surk, the red sandal. So they got me 2 leaves. The sandal, red sandal, I don't know which one is what. This is, they say red sandal, and this is the Pterocarpus marsupium, the duplicate red sandal. They gave me the leaf. We extracted the DNA at our CCMB, and when we did the sequence, we found a 5 base pair insertion in the duplicate red sandalwood. So here we got, which we cannot identify with botanical or microscopic or chemical method, but we can do it with DNA method. So this is the beauty of this, which will give the ultimate identification. So you know here the important problem here is this: the red sandal is an endangered species and endemic for Telangana and Andhra, and it's used as, the bark is used as an antibiotic drug, which has a big medicinal value, and economically it's a very important plant species. So that I was telling the 5 species insertion here you could see in the Marsupium Pterocarpus. And also they got me another plant species called Ocimum gratissimum with one set of plant having a small leaf size. Both are in the same right age and plant, and the other one has a big leaf. They were asking, what is the difference? Both are looking as per their classification, botanical classification, are Ocimum and gratissimum. But when we looked at based on barcode, we found a single nucleotide change. That this might be one. So still it is under development. We have more barcode to develop soon. So this is like, see, a leaf size change and a plant, there is some differences there. So, but here we have proven at the nucleotide base pair change. Sorry, that's what I got to phase out this picture. So you can see the nucleotide has changed from one of the nucleotide to another nucleotide in Gracemum of big leaf size. And also we have now developed the barcode for Asparagus officinalis, which is a very novel species in the Unani system, and there's no any barcode is available. This will be submitted soon to the database from NRE, MSD, and CCMB, and also we made the barcode of Euphorbia try Tirukalli, which is being used as a wound healing plant, I think, if I'm not wrong. And also we made the another one. They got me Artemisia absinthium, a species which is found in Hyderabad. They got me. We developed the sequence, and when we placed in the hierarchical cluster and the phylogeny, it is not absinthium. It is something else. So now I told that please change the name. You have to give a new name. It will be specific to Hyderabad, specific to NRUMSD, and specific to Unani system. So we have to name it. So here we can identify it. We can find the adulteration and what is the nucleotide change, whether it is right or wrong, and also if it is novel, we can identify, we can name them, and we can publish them. And also hemidesmus is other species we made recently, and these are the ongoing work for development of barcode for Unani drugs, which is just started recently, and hopefully in the next 1 year time we will be having a lot of database developed and published in the Unani system. With this, I thank here. I would like to acknowledge people at NRIUMSD, the director Yunus Munshi, and Dr. Kasif Hussain, Muhammad Kasif Hussain, and Pratap the botanist, and my student at CCMB, Ms. Anusha. Shukran, Jazakallah khair, Alhamdulillah. Thank you so much.
Thank you, Dr. Idris.
I'll be very happy to take few questions.
Absolutely deeper scientific work.
Yes. That influence. Yeah. Okay.
Yeah, we can take a question. Yeah. Please.
Use the mic.
So for research purpose, we can use DNA barcoding to identify the plant. So what is the cost for one DNA barcoding, sir? Like if you want to send a sample for research, we are doing some research.
Cost, you mean?
Yeah, cost, sir.
It's very cheap.
Like,
When you compare with all these modern biology experiment cost,
is it mandate, sir, to go for DNA barcoding if you're going for
that's
research?
what now we are now emphasizing. Indian pharmacopoeia is emphasizing. Now Unani system should make it. See, you make the drug, you treat the patients without knowing whether it is the right species and the right plant. So if you have the barcode, now you found that this is the plant, it is giving a good treatment for vitiligo patients. Then tomorrow somebody should not make the same with a different species or a nearest species. It should be the right species, right plant. So if you have the barcode in your monograph, what you have developed in your finding, they will match with that, whatever like you are doing it in Delhi. Now somebody want to repeat this experiment in Sri Lanka. They found the same species in Sri Lanka. If they found and if they want to work with the same plant, by botanical classification, microscopic or chemical method, you will not know the right way. If I do the barcode, I will be offshore. This is the species what Hyderabad, the Delhi people are working. So I can work with the same now. Or else they will ask you the plant or the drug they will import and treat the right patients. Yeah. So it is like you are working with offshore that this is my drug, this is the sequence. After this, there is no anything. Yeah, which will be coming, the protein sequence, which we are also doing. Proteomics we are doing, and then metabolomics we are also planning, which is a high-end techniques. So we are now started with soon.
So this facility is available in which facility?
It is available now. They are making it at Hyderabad. We are doing this experiment. Tomorrow, if you want to do barcoding, yes, you can do at your lab, or you can do it. You can send the sample to NRUMSD. Soon they will have the facility there. And also, we are giving a lot of training program to all the Ayush Ministry doctors. We conducted DNA barcode workshop for nearly 3 workshops we conducted where doctors from Ayush like Unani, Siddha, Ayurveda, and homeopathy people came. They trained. Now they are doing them by themselves. One example is Siddha Hospital in Chennai. They came to Hyderabad. We trained them 1 week training program. Now they got a project worth of nearly 1 or 2 crores, and they are now validating barcode for all the plants of Siddha system. So we are very privileged and we are very happy that the reason for which we are doing this has been achieved.
Okay,
so you can do it. It's not a big deal as the method is so simple.
That's why I wanted to know the cost, sir, because
cost is like, if you have to say, I may be wrong. If one plant from DNA extraction to sequencing will cost you some 5,000 to 10,000 Indian rupees. It's very simple. But yeah, the challenging here is getting the right sample without any what to call adulteration or contamination. Because whenever you collect any plant material from the soil or from the any like any from any garden, along with the plant sample, you will have so many other things like fungus will be growing in them, bacteria will be there. So you have to clean it up properly because this signature can pick anything. When I was doing that one species I was showing it is contaminated. It is contaminated with one fungus. Because contaminated their fungus means you can say, sir, it's a plant and it's a fungus. We can easily identify. No, for me, it's that AGTC matters. Fungus also will have the same AGTC as the DNA code, and plant is also having. So I get the different. So we have to get the right thing, and then the most challenging under the molecular technique is extracting the good DNA. Because plant cell, as I showed you, it has a cell wall, but as a human cell is just a plasma membrane. I can easily break the cell of human cell, but plant cell to break, we have to have a different method. Once you extract good DNA, you can do complete genome map of that particular plant, which is also under development. We are doing some few species we are targeting to do a complete genomic sequence and all their genes which are expressed in specific to Unani system, how they are doing. Yes, sir.
सर एक सवाल मेरा भी है
Please, sir.
विलेनिया सोमनीफेरा असगंध यूनानी लिटरेचर में असगंध नागौरी को रिकमेंड किया जाता है। नागौर से जो पैदा होती है सबसे अच्छी क्वालिटी मानी जाती है उसकी। आपने कौन सी क्वालिटी ली है, कहाँ से ली है, क्या अगर कंपैरिजन करेंगे क्या?
नहीं, मेरे को ये इंडियन फार्माकोपिया कमीशन गाजियाबाद से आया था पाउडर फार्म में। उनको चाहिए था सीक्वेंस क्या है, इसका बारकोड क्या चाहिए, बिकॉज दे वांट टू अमेंड इन द मोनोग्राफ। वी डोंट नो एनीथिंग। वो विटानिया सोमनीफेरा प्योरेस्ट फॉर्म में they collected from the market, I think, because you know the system, how Indian Pharmacopoeia Commission works. That's the main place where all the drugs are being, library is there, reference standard is there, sequence there. They develop the monograph. Every year they come up with this voluminous report. For the first time, it is 2022 or 2021, they came with the DNA barcode for our own herbal drug plants.
सर एक स्टडी ऐसी भी होनी चाहिए जहाँ का मेंशन किया गया है कि ये क्वालिटी यहाँ सबसे अच्छी मिलती है।
हाँ, वो भी है, वो भी कर सकते हैं।
It is
there. Because when we were doing this, like simple, collect neem from Assam, Gujarat, Kashmir, and Chennai. You will find a lot of variations. Geographical polymorphism you will find out. Then we can do. This is the study. Okay. You got a project now. Apply right and you get money. So this is the new next step of identification, classification, and a way we can achieve the right way to right plant, right drug.
Right. Actually, it is so simple. When you buy any allopathic drug, you'll find what? Whether it contains that particular
Yes.
one, the expiry date is there,
Yes.
or a right component is there. Likewise, if you are using Unani or Ayurvedic, wherever you use a plant one, you have to be confirmed that you are using a right species.
My aim, that's what we should have.
That is the importance of a barcode.
Yes, we should have that aim when we get the drug. It should be the sequence should be there in the drug bottle, along with the other specifications. Yeah.
Sir.
Yeah, I'm Dr. Kashif. Actually, I would like to add something which Dr. Arshia ma'am asked and Mustahsan sir. So initially we have started the work because we are using this barcode as a tool actually. So I am working as a botanist there, and taxonomically we can identify the plants whatever we are cultivating in our herbal garden or something sources we know. But as far as the efficacy of the drug, the authenticity and identification is very much important. And you know that when Dr. Zakir is here, he's running this pharmacy section, and there we have a drug identification committee for that. So in that, pharmacognosist, botanist and chemist and Dr. Zakir itself, one chairman is there. So we face a lot of problem during the identification when we purchase the drug from the market in bulk quantity. So we have to check whether, if you are, suppose you are taking the plant of Stereocarpus centralian, let's say wood, okay, so red color they will base, and how can you identify that? So in powder form, though, when you are going to pharmacognostical studies and investigations, there are some limitations in that. So this as a tool, and Dr. Mustahsan sir has asked a very genuine question that Nagori is there, and your Sena Makki. So like that we can go further for this, and in Unani Pharmacopoeia there is a lot of contradiction is there.
Yeah.
So we can also go for that, because for single aim, if myself is Dr. Kashif and you are identifying me as a Zakir, as an Idris Saab and Pratap,
Like that.
so how can you identify myself? So I have to show my Aadhaar card there at the gate.
Answer is barcoding or this one.
Yeah, this one. Yeah. So it's a madam. It's actually a tool, and now it's a age of AI and machine learning. You have a lot of these speakers on this side, and
Actually, barcode is nothing. It was the God who created a human structure, human creature
The
of
living
human
structure.
below.
Yeah.
They start with this barcode.
Yes.
So it is there. We are just validating that.
So that's why Idris sir is already saying that he only concerned with the 80 CG, nothing else. He
The
doesn't
nuclear
know
for
what is
base
Unani, what is Ayurveda, what is Siddha.
Yes, yes.
He is
And,
specifically talking about the plant itself and its proper.
I can't even pronounce the plant's name properly. It doesn't matter for me.
Okay.
And now coming to your question, madam. One India, one Unani family. So contact Hyderabad people, get trained yourself. You do this. Don't send your sample. You can do it in your lab. It's a minimal requirement is needed.
I am from clinical department, sir.
Madam, you won't believe, one clinician, a few clinicians from Siddha hospital came. Now they are doing it. You don't think molecular biology is a very tedious job. Any clinician, any biologist, any Unani doctor can do it.
Because we go for publication of our
Yeah.
research cases, na, sir. So usually now everything is, they're asking validation, authentication, everything. So better we go for a DNA barcoding. It will give a plus point that whatever species
Yes.
we have taken.
Yes.
It is.
If you submit with the barcode, you can submit it to a good journal with good impact.
And it is very useful for the single drug. When you are
That's
using
what
the
I'm saying, for
single
single
drug,
drug.
so there is no formulation at all. So that will specifically work.
If there is another procedure for mixed sample,
I think we will discuss
Yes.
during a tea
mixed
time.
sample if it's there,
It
in
is
the next,
already
inshallah, in the
10:50.
next
We have
Unani
to cover another
day will
three.
cover.
So thank you very much.
Thank
Dr.
you.
Idris.
Thank you so much.
Interesting one.
Thank you so
Interesting
much.
one.
Thanks everyone for your kind attention.
Yeah. Thank you. I'll request my colleague Dr. Morgana to introduce our second speaker, who is Dr. Mohammed Rasool. Please, ma'am. He's here. He's here.
Very good morning, ladies and gentlemen. So we'd like to welcome Professor Dr. Mohammed Rasool, a distinguished professor of immunology.
Sorry to disturb, I'm not Mahbub Khan Rasool.
Okay. All right, so we welcome Professor Dr. M. Rasool, a distinguished professor of immunology, currently serving as the Director of Sponsored Research and Industrial Consultancy at Vellore Institute of Technology, India. His research focuses on unraveling the molecular pathogenesis of autoimmune-mediated arthroidal disorder, rheumatoid arthritis, while integrating potent novel therapeutics into RA disease management. He has authored 112 research articles in international journals with a Google Scholar H index of 43, I-10 index of 91, and total citation of over 4,000, along with a cumulative impact factor of 354. So to date, he has completed 7 research projects exceeding 2.5 crores. His contribution to drug research in Unani medicine earned him the Best Research Paper Award from the Ministry of Ayush. He's been an invited speaker at various international and national forums across the world. He holds a position as a visiting faculty member in Turkey. Along with his many accolades, he received a Lifetime Achievement Award from the Society of Chemical and Synthetic Biology India. His outstanding contribution have placed him in the world top 2% scientists by Stanford. So Professor Dr. M. Rasool, the stage is all yours. So today he will be presenting his topic, his top title, Majun Chuchini inhibits dendritic cell activation via increased PD-L1 expression and reinvigorates the Th17 tract balance in the treatment of RA. Over to you.
Okay, thank you, chairpersons, for introducing me. First of all, I'd like to thank Director General Dr. Zahir Saab for inviting me to this international conference to share some of our data to you people. Okay, so today I am going to share some of our research findings, nothing but the Majun-Shopshini, how it inhibits the dendritic cell activation via increased PDL1 expression, and how it is stabilizing the TH17 and Treg imbalance in the treatment of RA. Okay. Say RA. RA is an autoimmune disorder characterized by the inflammation of synovial joints. All of you know that once a person, if he got RA, he can't move from freely from he can't do any normal life. So all of us as researchers, we used to think about only the cancer and other infectious diseases like HIV. But you have to remember that once you got the disease like HIV and cancer, it will kill you, but RA, it will take your life. It won't kill you, but it will take your life. It won't allow you to live a normal life. It will keep you to be as an observer. Okay, you can't be in 11, you have to be as a substitute, okay? You have to be in the bench. So nobody will like to be as a substitute. So as a researchers, it is our duty, it is our responsibility to provide awareness as well as to provide any therapeutic intervention for the possible management of this disease. Okay, when you are coming to this distribution, around 1 to 2% of the world population has been found to be affected by this disease. In this around women are more susceptible to this disease when compared to the men in the ratio of 3:1. Okay, so when you are coming to this etiology, the etiology of the disease is still unknown. That is why it is very highly complicated. Not only this RA, most of the autoimmune disorders, if you are saying there is no proper reason, that is why there is no proper complete cure. So we are having the medications only to control the management of these autoimmune disorders like RA. So when you are saying this pathogenesis, even though that etiology is not properly known, but a lot of research has gone through across the globe. They had given some some openings so that you can understand the pathogenesis of RA, so that we can try to control that disease. Okay, when you are saying this, slide, you can say that mechanism here. What are the possible reasons for the occurrence of RA means? You can say the genetic factors, environmental factors, and infectious triggers. Whatever the reason, okay, if there is any disturbance in the molecular structure of your self-antigens, okay, then what happens? The self-antigens will start to behave abnormal by your immune system. So your immune system will start to produce immune response against that autoantigens. What happen? First, the dendritic cells, which is a most important immune cell present in your blood. What it will do? It will trap that self-antigens and it will go and present to the T cells in the secondary lymphoid organs. Once it is presented to the T cells will undergo differentiation to TH17 and T follicular helper cells. The TH17 cells, what it will do? It will secrete one cytokine called IL-17. This IL-17, what it will do? It will activate the synovial lining, which is the major component of your joints. Okay, the synovial cells, once it will undergo activation with the help of this IL-17, and then it will release one protein called rankle. This rankle, what it will do? It will go and bind to the one precursor called monocytes, to the ligand called rank. This rankle will go and bind to the rank in the monocyte. Then the monocyte will be converted to macrophages, and then at last to the osteoclast. That osteoclast, what it will do? It will start to release some enzymes that is the cartilage degrading enzymes, which will ultimately erase the bone. Okay, so if you are not controlling this immune response, it will end in the bone erosion, which will cause the bone deformity. In addition to this TH17 cells, one cell has been formed during the differentiation of T cells, that is nothing but T helper follicular cells. This T helper follicular cells, what it will do? It will secrete a cytokine called IL-21. That IL-21 will activate the B cells, and then the B cells will will produce the antibody. That antibody will form a complex with that antigen, self-antigen, to form antigen-antibody complex. Once that antigen-antibody complex is formed, then what will happen? The complement proteins will get activated. That complement proteins will undergo activation to produce various inflammatory mediators. These inflammatory mediators will maintain that inflammation in that area. Ultimately, what will happen? That synovial cells, which is present in that synovial joints, it will undergo activation, which will release that various metabolic hydrolytic enzymes. That hydrolytic enzymes will degrade the bone, which will end in bone deformity, which will cause the bone destruction. Now the question is, this is the most accepted mechanism for the pathogenesis of RA. Okay, most accepted immunological mechanism, which has been proved. Okay, now the question is how we can able to target this presentation of self-antigen to the by the dendritic cells to the T cells. Okay. So if you are disturbing this presentation of the self-antigens by the dendritic cells to the T cells, then the T cells won't undergo differentiation to Th17 cells and then T follicular helper cells, so that we can control the pathogenesis of RA, so that all these reactions will be stopped. Okay. So we did this. So we had this question in our mind while we are framing this hypothesis. Okay. So this is all the treatments which is currently available in the market. So various inhibitors. So when you are seeing all these inhibitors, so which has been targeted to various immune response, what I had shown. But one thing you have to remember, all these inhibitors are, it is widely used or widely possible by only the high sophisticated peoples in abroad. Okay, it is not available, it is not affordable by the common people like us. Okay, like in developing countries. So what we can do? Okay, so what we did for the past around more than 15 years, we are working in our lab to discover various natural medicines which can be able to disrupt the immunopathogenesis mechanism. What I have told. Okay, so this is all the active compounds what we have screened, and then we have found that it is found to have that immunological regulations against this pathogenesis. Okay, so in this research, we came across one Unani drug called Vigyan. So one thing I want to tell you, we are working on various active principles, but we are ignoring the traditional medicines like Ayurveda, Siddha, Unani. See, unless until you are not as a researcher, as a basic researcher, if I am not giving importance to my natural resources which is present in my country, okay, if it is not proven, it has been used by the physicians for thousands of years, but there is no proof. That is why we are not getting any proper respect among the Western people. If you are not giving a scientific proof or scientific validation, how you can expect? If you are not publishing that studies in the very good domain journals, then how you can be able to get the attraction from the researchers across the world? So this question came across, then we searched, then we proposed this hypothesis, why we should not work on this Unani drug, which whether it has been what it has been used for this treatment of this arthritis. Okay, we thought whether this drug is having the ability to control the immunopathogenesis of RA, and then most importantly, we had the question that whether it can able to block that presentation of self-antigens by the dendritic cells to the T cells. Okay, with that question, we proceeded with this Majan Chowshini. Okay, so we started this evaluation with this FLS cells. The FLS is nothing but it is a main compound of your synovial joints, the fibroblasts like synovicites. This Majan Chowshini was found to inhibit the proliferation of fibroblasts like synovicites. As I told you, the synovial cells, if it undergoes activation, it will release the rankle. Okay, so if you are inhibiting the activation of the synovial cells, and then if you are inhibiting the secretion of rankle from the synovial cells, then that is a good strategy for the treatment of RA. We found that this Majan Chowshini is found to have that ability to inhibit the proliferation of FLS as well as that secretion of the rankle. That this findings has been published in on journal. Okay, and then with this promising result, we take forward for our main objective. Okay, this is one of the groundbreaking result what we have identified first time. Okay. You won't believe, we are the first group, we have published, we have reported that this Majan Chowshini is having the ability to upregulate the expression of PD-L1, negative immune checkpoint inhibitor, which is present in all your T cells. Okay, PD-L1. Okay, so just how it is possible, we put this question, whether this Majan Chowshini is able to block the dendritic cell maturation and restore that PD-L1 expression that corrects Th17 and Treg imbalance. Before going to this objective, I want to explain you for 1 or 2 minutes. Okay, what is the importance of upregulating that PD-L1 expression? All of you know that PD-L1 will get expressed in the dendritic cells, especially when it encounters the cancer antigens. Okay, this PD-L1, what it will do, it will go and bind to the PD-1, which is expressed in the cancer cell, which is expressed in the T cells. Okay, so when the PD-L1 go bind to the PD-1, which is expressed in the T cells, it will block that this binding will block the T cell activation. Okay, so that the T cell won't undergo activation, and then that will be a good strategy for the good news for the cancer cells, so the cancer cell will grow. That is why in 2018, Japan scientists, they received that Nobel Prize for discovering that monoclonal antibodies against this PDL1, but here the mechanism is reversed in the case of autoimmune disorders. We want that PDL1 expression has to be upregulated in dendritic cells, because the PDL1 expression is get upregulated. What it will do, it will stimulate the expression of PD1 in the T cells, so that when the dendritic cell go and present that self antigens to that T cells, this PDL1 will go and bind to the PD1 in the T cells, so that what will happen, dendritic cells will, what the PDL1 and PD1 binding, it won't allow that T cells to undergo activation. The T cells won't undergo activation, it won't undergo T cell differentiation, and then it won't form that Th17 cells as well as the T helper follicular cells. Okay, so that is the question what we put, whether this Majun-Chop is having the ability to inhibit this, whether it is having the ability to upregulate the PDL1 expression and inhibits the T cell activation. Okay, so this is the strategy, experimental setup what we have used for evaluating our drug. We established the authorities in the animals by inducing adjuvant mycobacterium tuberculosis, heat-killed mycobacterium tuberculosis, okay, that used to establish the arthritis. Okay, after establishing the arthritis, what we did, we allowed the animals to have the arthritis up to 11th day, and from the 11th day, we administered the Majun-Chop up to 20th day, and then 21st day, what we did, we excised we sacrificed the animals, and then we excised the spleen, okay. And then after excising that spleen, what we did, we went for dendritic cell population. Okay, so in that spleen, just you carefully listen. Here only we are going to see the important data. Okay, you can see in the second panel. Okay, in arthritic animals, that the dendritic cells where we have isolated that from the arthritic animals, you can see that dendritic cell maturation is very high. Okay, that means that dendritic cell, it is what it trapped the antigen, and then so that it is getting activated. Once it got activated, it will express the surface markers called MSC class 2 and CD86. We measured the CD86 and MSC class 2, and then we came to know that dendritic cell has got activated. And then whereas in this Majun-Chop treated drug, this MSC class 2 and then CD86 and MSC class 2 has been found to be downregulated, which shows that Majun-Chop is having the ability to inhibit the dendritic cell maturation, that is activation. If that maturation is inhibited, it won't be able to present the antigen. Okay, and then PDL1 expression. Here is a very important data. Okay, you can see that in arthritic animals, the PDL1 expression is found to be downregulated, because during the time of any activation happens, okay, that the PDL1 has to go down naturally. Okay, then only it can go and present that antigen to the T cells. Okay, since the problem is here the antigen is ours, so that the PDL1 expression has to be there. Okay, it should not go downregulated. As suspected, in arthritic animals, the PDL1 expression is found to be downregulated, whereas in the Majun-Chop the PDL1 expression was found to be upregulated. Okay, and then the Th17 differentiation. Once the PDL1 and PD1 binding is inhibited, we want to check whether the Th17 differentiation has been controlled. As we expected, we came to know that this arthritic animals, the Th17 differentiation is more, because the dendritic cell activation is more, so that the Th17 differentiation is more. Whereas the Majun-Chop treated drug, the Th17 differentiation came to low, which gives an indication that dendritic cell maturation is downregulated, it is disturbed, so that the Th17 differentiation has came down. And then we checked this IL-17 cytokine, the expression of IL-17 cytokine in the synovial tissue, and then we confirmed that Majun-Chop downregulated that expression of the IL-17 when compared to the arthritic animals, which proves that, which gives an authenticated support that it is able to inhibit the dendritic cell maturation, so that the Th17 differentiation is less, so that IL-17 cytokine's expression is less in synovial tissue. And then we checked the Treg cell differentiation. Once the Th17 cell came down, Treg cell has to go up. Then only the immune balance should be there. Treg cell is responsible to producing a cytokine called IL-10. The Treg cell should be high in case of the autoimmune disorders. Then only it will release the anti-inflammatory cytokine called IL-10, so that the IL-10 will go and suppress that Treg cell differentiation. Okay, as we expected, the Treg cell, the Treg cell differentiation is low in arthritic animals, whereas the Majun-Chop upregulated this Treg cell differentiation. One thing I want to tell you here, when any drug, if it inhibits the Th17 differentiation, automatically the transcription factor, the STAT3, will get switched over to STAT5. Okay, that STAT5, what it will do, it will upregulate the expression of the transcription factor, which is responsible for what is that Treg cell, FOXP3. Okay, that FOXP3 has been found to be upregulated in this T cell, which indicates that population of Treg cell. Okay, and then we checked the expression of IL-10. The IL-10 is the cytokine which is released from the Treg cell. The Treg cell is get upregulated. IL-10 should be found to be present high in the synovial tissue. We came to know that Majun Chobshini increased the expression of the IL-10 in the synovial tissue, which indicates that, which supports that our previous data that Treg cell is more in that directed artery animal. And then this is the mechanism what we have for investigated to check what is the reason for the PD-L1 expression, because the time is limited. So I will skip this, and then just you can see the data how that the FOXO1 is responsible for the upregulation of PD-L1 during the what disease condition, arthritic condition. The FOXO1 will undergo a phosphorylation, and then it will undergo translocation to the cytoplasm. It will get degraded. Okay, and then this is the FOXO1 expression which got upregulated in the Majun Chobshini treated animals, so that which is responsible for upregulation of PD-L1. Okay, and then we checked in vitro also. Okay, what we did, we isolated the dendritic cells from the animal, and then we activated this dendritic cell with the expression of LPS, and then we checked that PD-L1 expression is found to be low in that LPS treated dendritic cells, whereas the Majun Chobshini treated dendritic cells, that PD-L1 expression is found to be high. Okay, which shows that it is having the ability to increase the upregulate the PD-L1 expression. So when this dendritic cell is presenting the antigen LPS to the T cells, this PD-L1 will increase the expression of PD-1. Okay, so that this binding will stop the T cell activation. Okay, and this is the mechanism what we have proposed. Okay, just we can say since chairpersons are forcing me to stop. So
time is over already.
Yeah. So
We
no.
are not forcing.
Just in later manner I am telling. Okay, so Majun Chobshini enhanced the PD-L1 expression. Okay, that abolished dendritic cell maturation via regulation of A3, AKT, FOXO1 axis, thereby hindering TS17 cell differentiation rheumatoid arthritis. Okay, so this study provided a scientific proof that Majun Chobshini is promising candidate for the treatment of RA. Okay, and then this study, it has been published in Cytokine and Immunology high reputed journal. And then the candidate who worked in this project, now she is a postdoctoral scientist in Harvard Medical School. Last month she got a paper in Natural Immunology. Okay, it is because of the Minister of Ayush who trained this girl to go to this level. So in this forum, I thank the Minister of Ayush for giving this opportunity to her to get trained in this immunology. Okay, and then this study has been reported. It has been included by the daily magazine Indian Express, and then it has been published. Okay. This is our lab team. Without them, I may not be without by their hard work only I am here. So it is my duty to thank them for their hard work. Okay, and then I thank all my funding agencies who are all responsible for making my research meaningful. Okay, and then useful to the society. Thank you.
Thank you, Prof. Rasul. Prof. Rasul, you have shared a very interesting pathway. Now, one of the latest therapeutic target is the manipulation of tolerogenic dendritic cell to suppress the immune system. So, would you say Majun Chobshini has a similar activity?
Just we have to check. Very good question. I appreciate your recent update. Okay, the tolerogenic dendritic cells. We have not checked in that angle, so it may have the property to increase the expression or increase the population of tolerogenic dendritic cells. We had that question to check whether it is possible to upregulate the expression of PD-L1 only. With that angle only, we investigated this study.
Right.
Did you try IL-6 also?
IL-6, it is a more common cytokine, so that we checked that. The TNF alpha, IL-6, IL-beta.
You use CFA only.
We use CFA only, complete sponsored joint.
Thank you very much.
Right. Does anybody has any questions?
I think we will take up it during the tea time.
Okay, thank you.
Actually, I am not pressing you. The organizing.
No.
I am just
See,
transmitting to
what
you.
I want to
Thank
say,
you very much.
this is not the study which can be explained in 15 minutes.
Yeah.
Okay,
Good.
thank you. Thank you all.
Thank you.
A very good morning to one and all. Good morning to one and all. Let me introduce our third speaker, Tanveer Naved. Dr. Tanvi Naved is a distinguished academician with over 2 decades of experience in teaching, research, and administration. He currently holds the position of Deputy Dean at the Faculty of Health and Allied Sciences, MET, University Noida. Known for his strategic planning and execution skills, he has played a key role in organizing major events, including the 65th IPC in 2013 and 70th IPC in 2018, and a recent international conference at MET University. Dr. Naved is a recipient of University Gold Medal. He has a strong research background with 75 publications in reputed journals with high index factor, and he has a mentorship experience of having 12 PhDs and 30 master students. He is he actively participates in professional organizations such as IPA, IPG, etc. And he is a member of expert committee of World Health Organization, WHO, and a National Medicinal Plant Board under the Ministry of Ayush, Government of India. So over to you, Dr. Naveed. The floor is all yours.
Thank you very much for such elaborative introduction, ma'am. Assalamu alaikum and a very good morning to all. Before I start my presentation, I just like a disclaimer that my presentation is not the original research work, but it's sort of review of some of the research carried on the adaptogenic or the immune boosting plants as well as the formulation particularly used in the Unani system of medicine. So though it's just a review of last 4-5 year research work carried on this plant, we can say in the post-COVID era. And as Dr. Surendra Singh sir has already instructed that the tea break is around the corner, so I sure to complete in next 10-12 minutes time so that we all have a good tea break after one more presentation. So I think this initial slide I can skip because we all are aware of this thing. The particular reason for choosing this topic, because I could have share some of the research finding of one of the project on Dawal Kurkul which carried on MET University, in which I am the co-PI. But I deliberately choose this topic because though the God grace all the severity of the COVID has been almost get rid of, but unfortunately those who have suffered, those infected during the the first wave during the March April 2020 and subsequently more dreadful wave in the April 2021 is still they are not come out of the symptoms, and there is a prevalence of the symptoms and the reoccurrence of the many of the symptoms among them. So I'll quickly, discuss some of the well-known Unani formulation on which the research has recently carried out to their pivotal role in the curing of the COVID-19. So one first preparation is the Tiryak Arba. Tiryak is basically the antidote preparation in the Unani system of medicine, and this formulation is consist of Commiphora myra. Commiphora is known for its anti-inflammatory effect. You can, the commiphoric acid is a well-known anti-inflammatory agent, and along with it also contains the Laurus nobilis. And basically the drug, this formulation, Tiryak Arba, is traditionally used in the respiratory symptom ailment. So again, if you correlate with the COVID, there is a severe, respiratory ailment occurred during the COVID, those who suffered the severe infection of the COVID. Now the next formulation is the Rogan babuna. Babuna is otherwise is this chamomile flower. It's known for its ardent, beauty, and you perhaps aware of the various, green tea formulation with chamomile and other, but here the Rogan babuna is again known for its anti-inflammatory effects, and also in the nasopharyngitis condition where the inflammation of the nasal passage, so, and also for the sore throats. Again, the third formulation is Arke Ajib, which is basically a combination. You can see that thymol, menthol, and camphor. In general, they are the refrigerants, give the cooling effect. But at the same time, the formulation is known for the hepatic infections, and also they are using in the Nazla Wahhabi, which is a swine flu. Swine flu, that generally is, infected by the pigs. So this is the another formulation. Now I'll then, yeah, Khamira Banafsha. Banafsha, I think some of the students are sitting and pharmacognosy, they know that over the years, the Banafsha is one of the finest remedy for the cough and cold. The Viola odorata, beautiful flowers. But at the same time, the recent, again, the studies focus on the respiratory ailments. And that's why the main reason of, covering all this formulation, because somewhat they are, good for health and good for the respiratory tract system, especially the bronchi. And also, this is another formulation is the Lahuk Sapista. It's a well-known semi-solid formulation, and Lahuk Sapista Kayar Shamri is very good combination of Amaltas and the Sapista. So this is another formulation. Further, Sharbat Sadar is one of the popular formulation. I think many Unani, experts are sitting here and they are well aware about the wonderful, beneficial effect of Sharbat Sadar. Again, you can see that it is used in the common cold and the respiratory disease. So it contains the Adotota vasica, which is also known for its bromhexin sort of effect, which is good for the bronchi and the respiratory tract. And again, one very popular Unani formulation is Khamira Marvarit, which is again Khamira, all are semi-solid sugar-containing formulations, and it is also known to help boost the immune system. And one very important is the Sapoof-e Azgand or the Azgand Sapoof. Azgand is the Ashwagandha, the Withenia somnifera. In the subsequent slide, I'll give you a very detailed mechanism of action of this drug, so I'll skip here. And other one is the Habbe Bukhar, so which is otherwise used in the various type of fevers. Now coming to the first plant, I'll discuss very briefly about the plant, is the Withenia somnifera Asgandh or Ashwagandha, which is a wonderful plant. We are all aware about the wonderful, but one slide. Dr. Singh sir, this slide actually I have taken from one research paper which was published, probably the first paper during the COVID time or immediately after the COVID time by the IIT researcher in collaboration with some Japanese researcher. So the paper actually highlight the role of Asgandh or the Ashwagandha in the treatment of the COVID, and how. Because this paper suggests that once we take the vithanone, which is an active phytoconstituent of Withenia somnifera, and propolis, which is obtained from the V glue, a very simple plant. The combination could inhibit this main protease, the AMPRO, which is probably the enzyme responsible for the multiplication of the coronavirus. So this is one of the paper, I have taken the reference from the IIT researchers paper along with the Japanese paper. Now quickly I'll go through all the plants, sir, so because it will save the time also. Gloe, we all know it's a wonderful plant, the Dinospora cordifolia, and the whole part, whether it is stem, root, or the whole plant is known for its wonderful activity, including the aphrodisiac activity, immunomodulatory activity, antioxidant activity. Moringa, I think my precedent speaker are from basically from the southern part of the India, and sir, you are aware that without the the drumstick, no culinary, no nothing is possible, is complete. But it's a wonderful plant. I've seen many researchers, uh, advocating the use of moringa in the daily intake, and it's a very easily available. Otherwise, very fond of this.
The problem is the toxicity as well.
So ma'am, एलोवेरा के तो क्या ही कहने हैं, उसको तो स्किप ही कर देते हैं. एक ये अच्छा प्लांट है यूनानी में, फोवा. So it's for basically the leaves has been used for in various Unani formulation, obtained from the Rubia cordifolia. Then kalonji, again a very good hepatoprotective drug and very easily available. ये तो वो फॉर्मुलेशन है जो किचन्स में भी अवेलेबल होते हैं, घर के रेफ्रिजरेटर में भी कुछ चीज़ें आपको मिल जाएंगी. सो दैट्स वाई, बट व्हेन यू कम टू देयर pharmacological effect, their remedial effect, you will find that in most of the Ayush formulation, whether it is the Unani formulation or somewhat the Ayurvedic formulation, they are present. Like Amla, one of the best example. जब हम त्रिफला की बात करते हैं तो सबसे पहले आंवला आ जाता है. And amla worldwide known for its wonderful antioxidant effect. Gural, the hibiscus plant, it has a very good property even in the preventing the hair fall and the problem related to the hairs. क्योंकि आप मार्केट में देखते हैं कि बहुत सारे shampoos, anti-dandruff, preparation, they are available. So these are some of the plants which are used as a very versatile plant. The curcuma or the haldi, well known for its anti-inflammatory effects. Just to take 1 minute, sir, because some of the students are sitting from my department also. Other day I was, teaching them this particular drug, and I was giving one example of Mahabharata and Ramayana serial where we observed that कोई भी अगर सैनिक घायल होते थे, so there was the old proverb कि इनको हल्दी का लेप लगा दो. And I think it was 1977 or 78 where all the curcumin's effect was come to known to the all the worldwide. This is the responsible phytoconstituent for all the anti-inflammatory effects. So, but because you can see from the ancient time, it was all the prano mein aap ya charak samhita, everywhere you find the wonderful effects of curcuma. Neem, again, wonderful plant known for its purifying effect, the antibacterial effect. And this ficus, bargat, one of the wonderful plant and known for its, very quickly you can see the hepatoprotective, anti-helminthic, anti-proliferative and anti-inflammatory effects. Then muesli safed. These days, muesli is being used as a wonderful nutraceutical. You can find the various, all this Kellogg's products and many others. Safed muesli. It is also a very good aphrodisiac also. Then satavar. This is a wonderful plant. Very quickly, I like to mention one of the major use of satavar. It has a galactagogue properties. Galactagogue basically the milk enhancing. So in some of the mothers after the childbirth, they could not sufficiently feed the infant. So for increase the secretion of milk from the mammary gland, the formulation of Satavar is used in tradition by the many physicians. Then this is again lotus flower. Then glycer has a glabra, one of the best remedy available for the sore throat.
Dr. Naveed, if you could just.
Yeah, ma'am. I'll complete in 1 minute. So very quickly I'll go through because the Piper longum is known for this wonderful effect as well. Then we have the Halela, Papita, Amla, of course, we all are aware. Then Isabgol well known for its anti-constipation effect. Then Babuna, already we have discussed. We all aware. And last but not the least, the Ocimum centum. I think plant तुलसी जब एक कान में पड़ता है तो उसकी एक सेक्रेट भी हमारे अंदर जज्बा आता है और उसका medicinal value भी आता है. With this, I like to thank all the research, the chairperson and the audience for their patient learning. Thank you very much.
Thank you, Dr. Naveed, for giving an insight on immunomodulator medicinal plants. Thank you so much.
Shukriya.
I'll call upon the last presenter, Dr. Amisha Vora. She is a faculty at SPP School of Pharmacy and Technology Management. I think it is in Narsi Munji, Mumbai, na? Yeah. She is a PhD in Pharmaceutical Sciences from Narsi Munji, Mumbai with a vast teaching experience of 20 years. And she is expert in designing of in vitro assay and bioanalytical studies. Also involved in a preclinical study of Ayurvedic and Unani formulation. And till now, she has guided 40 PG students and many PhD students, and supervising the evaluation of natural product which can be useful in Alzheimer and chronic kidney disease. Dr. Amisha.
Thank you, sir. Thank you very much. I feel honored and privileged to be presenting here today on the occasion of Unani Day 2025. And I would like to thank the entire organizing committee, CCRUM, Ministry of Ayush, Government of India, for giving me this opportunity to present my research work today. Thank you. So it's a very small presentation. It will not take too long. It's on the neuroprotective potential of this polyherbal Unani formulation, marginal Nisyan, in amyloid beta-induced animal model of Alzheimer's disease. So we all know about Alzheimer's disease is a progressive neurodegenerative disorder, which is marked by the degeneration of cholinergic neurons, resulting in cholinergic deficit and resulting in cognitive decline and memory loss. In 2019, India had estimated 3.6 million active cases of Alzheimer's disease and other dementias, and about 8.8 million Indians older than 60 years of age live with dementia. It's a complex disease with pathological contributions from multiple factors: aggregated amyloid beta protein and tau protein in the neurons of brain, extracellular in case of amyloid beta and intracellular in case of tau protein, are suggested to contribute extensively to the oxidative stress and inflammatory stress leading to Alzheimer's disease. So there is no cure of Alzheimer's disease. The presently approved FDA-approved molecules tend to alleviate the symptoms of Alzheimer's disease, but there is no cure of Alzheimer's disease. And there are polyherbal formulation naturally has multiple constituents. Each may act by different pathway, contributing towards contributed towards slowing down the progression of Alzheimer's disease and hence may be beneficial. So we have made an effort to check the effect of polyherbal Unani formulation, how it can help in the treatment of Alzheimer's disease. So basically this particular formulation has 5 constituents, 5 herbal constituents along with honey. Boswellia serrata, Acorus calamus, Cyperus rotundus, Zingiber officinale, and Piper nigrum, in the proportion as mentioned on the slide. We started our research work with the physicochemical characterization of the formulation as given in the Unani pharmacopoeia, and we found that all the parameters of physicochemical characterization were falling within the limits of Unani pharmacopoeia. We followed it up with the standardization of this formulation using the biochemical markers. We selected one from each plant, boswellic acid representing Boswellia, beta-azarone representing Acorus calamus, isoeugenol, 6-gingerol, and piperine. These are one marker from each of the herbal constituent, and we optimized the HPLC mobile phase to be toluene ethyl acetate chloroform glacial acetic acid in the ratio of 8:2 to 2.5:1. 8:2 This is our HPLC profile of the formulation. You can see the resolution of each biomarker in the HPLC. This is the HPLC densitogram. And this is the plate where we have spotted 2 concentration of each marker, 2 concentration of the mixture of each marker, and the 3 concentration of our Unani formulation being extracted mildly in methanol and concentrated and then marked here in the HPLC. We have validated the HPLC method with ICH guidelines, and we found that the linearity of each marker, the range fell within the range of 1 to 12 microgram per spot, the correlation coefficient being greater than 0.99. We can see the linearity and the standard deviation of intercept. The intraday and the interday precision was carried out by marking the spot of 10 microgram per spot 6 times for each standard, and calculating the RST value, percentage RST value was found within the limits of less than 2%. We also carried out the limit of detection and the limit of quantification for each biomarker, which was found to be, as shown on the slide, around within the range of 1 to 3 microgram per spot. So this is the HPLC work. We have also carried out the accuracy of the method, where the percentage recovery was found within the specified limit of 90 to 110% of the original amount. With the method, we quantified each of this marker in the formulation, and the quantities of each marker compound are written here in milligram per gram of formulation. So this is how we developed the method using HPLC and standardized the formulation with 5 biochemical markers. Going on to the preclinical studies in the Alzheimer's disease. So we had 7 animal groups with 10 animals in each group. We have done the administration of amyloid beta in the C1 region of hippocampus of brain. We used stereotaxic surgery to inject this amyloid beta in the animals. We had a control group, the sham control group, and the positive control, which was donepezil. Donepezil was given at the dose of 1 milligram per kg, and the treatment groups consisted of 3 doses of marginesia, namely 500 milligram per kg, 1000 milligram per kg, and 1500 milligram per kg, given orally for 30 days after the recovery period of stereotaxic surgery. So this is how our preclinical studies was planned. We went on to the checking of behavioral parameters at the end of our administration, and it was found that you can see that the escape latency significantly increased in the disease control group with respect to normal control group, and there was improvement in the behavioral parameters with the treatment group. Uh, though the results were not very significant with 500 milligram per kg of the formulation, however, it was quite significant with 1000 and 1500 milligram per kg of the formulation. Similarly, we proceeded with passive avoidance test, which is another test for the behavioral parameters. We found the similar results. There is decrease in the escape latency in the disease control group, while a significant increase was found in the treatment groups, as mentioned on the slide. The brain, after sacrificing the animals, the brain homogenate was prepared. We went ahead with the biochemical markers, the oxidative stress parameter markers, and the inflammation markers, where we have quantified the levels of glutathione and quantified the levels of MDA in the disease control group as well as the treatment groups, where you can see the similar kind of results as we have seen in the pharmacodynamic studies. Going on to the levels of acetylcholine esterase, which is a marker for Alzheimer's disease, and the neurotropic factor, the BDNF. Again, you can see the similar kind of results. In case of disease control, the acetylcholine esterase levels were significantly increased, and the BDNF levels were significantly decreased. The treatment groups improved the levels of acetylcholine esterase and BDNF, as you can see on the slide. We have gone ahead with the oxidative stress markers, TNF alpha, as well as one of the targets of Alzheimer's disease, that is GSK3 beta, and the NF kappa B, which also marks the inflammation. And we have seen the similar results as we can see on the slides. We have also gone ahead with the histopathology of the hippocampus region of the animal tissue. We have not stained A beta right now. We are halfway with this work, but this is just the histopathological parameters. We are further going to go with the sections in which we can particularly stain A beta and see how the A beta levels have changed in the treatment groups. So in summary, we performed the standardization of formulation using 5 marker compounds using HPLC. We studied the effect of this particular formulation at 3 dose levels, 500, 1000 and 1500 milligrams per kg. Based on our research, we draw conclusion that the formulation margenicyan at 1000 and 1500 milligrams per kg has acetylcholine esterase inhibitory, anti-inflammatory, radical scavenging, and A beta activities in A beta induced Alzheimer's disease in rats. We also conclude that margenicyan formulation shows beneficial effect in Alzheimer's disease by acting on the GSK3 beta NF kappa B pathway. So these are the key references here. We are thankful to the CCRUM for the project funding, which is given for the collaborative project where we are doing this formulation effect. And this is my research lab. I have 3 PhD students. All 3 of them are working in different areas. We are trying to set up the in vitro and in vivo lab for different diseases. So one of them is working on Alzheimer's disease, so she was the one GRF who was working on this project, and her PhD project is also lined up in case of Alzheimer's disease. We are developing the in cell culture lab also for Alzheimer's disease. The other one is working on chronic kidney disease, and the third one we are trying to develop the anti-malarial activity in our lab. Thank you very much for your time.
Thank you, Dr. Anisha. I think in all the funding agency, if you say DST, DBT, ICMR, the thrust area is either Alzheimer's disease or a Parkinsonism, and I think this type of study is a way forward, which may not be a potential one-go type of drug, but it can be an add-on type of this one.
adjuvant treatment.
We can take one question, yeah, and after that we'll break for tea. Yeah, please.
Thank you.
Use mic.
You can hear me.
Yes, I
I'm
can
loud.
hear.
No, it is
Okay.
It is all right.
All right. So you said like you chose the 3 doses, right? Did you check the LD50 first?
Yeah,
The-
the
the
safety
sec-
studies have been already published. So
Okay,
we
so
have
it
seen
wa-
the LD50.
So
Yeah.
it was the
And it has been published by him only.
Yeah, actually,
So
we have
Dr.
done the repeated dose toxicity as well. So up to 2000 mg per kg, there is no toxicity at all on this formulation. We have done on Majunisian itself.
You did what? Under OECD 423?
Yes.
We have done 407, yeah.
So my question was not initially jumped. So the question was like you use the adult mice or the old age mice, because Alzheimer disease, it's an age related disorder. Right?
Yes, 16
So-
weeks
so
old.
male and
We
female?
start male and female.
Both
Because yeah,
together?
both.
Okay.
Both the
All right. Thanks.
I'll make a point on to your question.
Mhm.
Uh, See, the prevalence of AD in a human being, it is a age-related one.
correct.
But we are not scrutinizing the animal from a AD colony. We are inducing
Inducing
AD
it,
actually. So it really doesn't matter whether it's a aged one or this one.
Yeah, you are right. But the thing is like there are more genetic altered mice.
Yeah, yeah.
APS mice.
Either you develop a genetic colony of that and you take out. That is the best model.
Exactly.
But in this case, it is induced by some chemical, and then we have taken that reference one.
Yeah, that's why my question was.
Yeah,
male
your
and female,
question
how
was
do you
right.
compare,
Question was right.
Yeah. Thank you.
So I thank you all speakers, Dr. Muhammad Idris, Dr. M. Rasool, Dr. Tanveer Naveed, and Dr. Amisa Hora on behalf of all my colleague, Dr. Mogana and Dr. Arzina. Thank you very much, and thank you.
Thank you so much.
Organizer.
Thank you, sir.
Jaldi
Now time has come to thank our speakers. We would like to present a memento as a small gesture of gratitude. May I request Dr. Mogana Sundari Rajgopal to present a token of appreciation to our first speaker, Mohammad Idris, Senior Scientist, Principal Scientist, CSIR, Hyderabad. Thank you, ma'am. Now I request Professor Surinder Singh to give the memento to Professor M. Rasool, Assistant Director, Vellore, Institute of Technology. Please come fast.
I publish around 30-40 people on RA. Yeah. I did an X-ray and this also. That is more useful.
I request
You are right. This cannot be changed.
I request Professor Tanveer Naveed to receive the token of appreciation from Dr. Arzina Jami. And lastly, requesting Dr. Amisha Vora to receive the token of appreciation from Dr. Musti Hassan.
Please.
We have a small token of appreciations for our chairpersons as well. Let's start thanking today's session's chair. I would like to present a memento as a small gesture of gratitude to our first chair, Dr. Mogna Rajgopal. Now I would like to call upon Mr. Anas Alvi to present a token of appreciation to Dr. Arzina Javi. I request Dr. Muste Hassan to present the memento as a token of gratitude to Dr. Surinder Singh. So once again, I would like to thank our chairs and all speakers for the wonderful, thought-provoking presentations. Now we will be having our next session after the tea break. Thank you all. Sir, सर हम सब सारे एक साथ करते हैं सारे चेयर सब एक साथ
साउंड चेक कर लेना भैया
नीचे तो जो हो रहा ही बहुत अच्छा हो रहा वो ना वो भी कुछ गए सेंटर पता नहीं बोलते विज्ञान वाले से कहीं तो यहाँ आएगा मैंने बोलते हम वाले तो डल जाएगा तो आपको लाइट चाहिए वरना तो फिर मैंने मूवी तो ऐसी फोटो एलईडी वाले इस पे डाले तो चले जाएगा विद्याम भवन वाले हेलो 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 माइक टेस्ट हेलो किसका बोलूँ मैं उन्हें बोलूँ कंट्रोल रूम तो बोलो ना बोलो
हेलो भैया साउंड चेक कर लो साउंड
नहीं मैंने दिया
साउंड चेक कर लो
अपने सर की कसम अब तो दे दो अरे तुम्हारे तेज हो यार अबे यार मैं अगर तुम्हें अगर अपने बंदों को मना करो यार अबे यार मैं सुबह से मेरे 600 में कर दिया अभी तो 5 बार पता चल आप लैब इसमें तो यार अच्छा चल रहा है ना दूसरा इसमें तो आईपीसी चल रहा है यही नेटवर्क इसमें भी
This isn't a laptop launch. This is a U launch. The new Lenovo AI PC has built-in AI that learns from you. In breaking news, the
BJP MP from Delhi and also former Delhi BJP chief मनोज जी बधाई हो आपको इतनी बड़ी जीत भाजपा को मिल रही है आप लोग सबका उस
वास्तव में दिल्ली की जनता का बहुत बहुत आभार है दिल्ली की जनता का बहुत बहुत धन्यवाद है और ये जीत नरेंद्र मोदी जी के गारंटी की जीत है मोदी जी ने ये सिद्ध कर दिया है कि अब मोदी जी के गारंटी पे देश के हर राज्य का विश्वास है महाराष्ट्र में भी ऐसे हुआ
थैंक यू बहुत सिंपल कर दिया
हरियाणा में भी ऐसे हुआ और कोई दो राय नहीं है अब नरेंद्र मोदी जी जब कुछ कह रहे हैं तो उसको एग्जीक्यूट भी कर रहे हैं और उनके एग्जीक्यूशन पे उनकी गारंटी पे प्रत्येक राज्य की जनता को विश्वास है दिल्ली ने सिद्ध कर दिया
जी एक सवाल और अभी क्योंकि अभी खबर आई है कि अरविंद केजरीवाल मनीष सिसोदिया दोनों हार गए हैं लेकिन गोपाल राय बहुत ही अच्छे मार्जिन से जीत रहे हैं तो इसका क्या संकेत है कैसे पढ़ रहे हैं आप इस डिफरेंस को
देखिए अरविंद केजरीवाल के हारने के बाद और कोई बात बची नहीं जाती अरविंद केजरीवाल तो ऐसा बोला करते थे नरेंद्र मोदी जी को चैलेंज करते थे कि इस जन्म में आप नहीं हरा सकते अरे अरविंद केजरीवाल जी आपको इसी जन्म में ऐसी हार मिलेगी और न जाने आपके इसी जन्म में कितनी हार मिलेगी आप लोगों के केवल ईवीएम से नहीं हार रहे हैं केवल वोट से नहीं हार रहे हैं लोगों के दिलों से हट गए नफरत है आपके प्रति ये मेरा मानना है और इसके लिए मेरा पूरा विश्वास है कि अरविंद केजरीवाल जैसी सोच कोई भी नेता रखने के पहले 50 बार सोचेगा
मनोज जी 2013 में आप एक जायंट किलर की तरह उभरे थे शीला दीक्षित को हरा के आज क्या आपको एक हिस्ट्री रिपीट इटसेल्फ वाला एक फीलिंग आपको क्या वो वाली फीलिंग आ रही है
चालू है
देखिए
कितनी देर तक
कहते हैं ना कि सबके साथ समय के साथ
ऊपर वाला
तो एक भ्रामक
ये टाइम रेडियो ऑडियो पे जा रहा है एक्सएलआर पे नहीं जा रहा है छुट्टी में डेट क्या है वो टाइम रेडियो अभी तो यहाँ से निकाली थी भैया आपका नाम यहाँ पे निकाली थी अभी नहीं रहना ये फ्रेम आएगा तो कैमरे को फ्रेम फिक्स करके अपना चलाओ जरा रुक जाइए देखना ऐसे भी नहीं आ रहा देख अर्थ ही नहीं आ रहा यार अर्थ तो पकड़ना चाहिए उसको आप अपने में लगा के सर के में तो लगा भाई अपने वाले में लगा दो सुबह तो इससे चेक हुआ ना क्या आउटपुट दे रहा है कि नहीं दे रहा है ये पता चलेगा ना लेकिन लैपटॉप आउटपुट नहीं देता लैपटॉप आपका वो
हेलो, नहीं आ रहा भाई?
लाख रुपया अब उस इनकम पे टैक्स नहीं लगेगा। तो मोदी जी को, निर्मला जी को इसके लिए बहुत बहुत धन्यवाद है। और यही विश्वास खड़ा करता है ना।
परवेश वर्मा अभी गृह मंत्री अमित शाह जी के घर पर हैं और वहां पर मुलाकात कर रहे हैं। अमिताभ तिवारी का एक सवाल है आपके लिए।
मनोज जी नमस्कार, अमिताभ तिवारी बोल रहा हूँ, बहुत बहुत बधाई आपको। 2014-15 में हमने देखा और 2019-20 में भी 2019-20 में भी कि भाजपा का एक सेक्शन ऑफ वोटर लोकसभा से आम आदमी पार्टी की तरफ शिफ्ट होता था विधानसभा में, और इसीलिए हम
Hmm? No, I'm not Professor Abdul Kavi, I'm Dr. Noman. He is sitting there, chairperson is there. Yes. I will start and invite the chairpersons and they will be inviting the speakers. Yeah, yes. Hello everyone, good noon and a very warm welcome to you all to this technical session on development of Unani Ahar through scientific approach. So as we are already running late, to start with this proceeding of this program, first I would request the honorable chairpersons of this technical session to the dais. Professor Dr. Abdul Kavi, Principal and Superintendent, State Takmiluddin College and Hospital, Lucknow, Uttar Pradesh. Dr. Abdul Kavi obtained the degree of MD in Immunology from Rajiv Gandhi University of Health Sciences in IUM Bangalore in 2010. He is working as acting principal and superintendent in the State Takmiluddin College and Hospital, Lucknow since December 2023 till date. He served as professor and head department of Ilmul Jarahat in Al Farooq Unani Medical College and Hospital, Indore. So Dr. Abdul Kavi has published multiple research papers and review articles in national and international journals of repute, book chapters and books. He was awarded by Deputy Chief Minister of UP for excellent work for yoga training arrangement on 21 June 2018 in Governor House, Lucknow, UP. So we welcome our first chair of this technical session. To welcome our first chair, I would invite Dr. Asma Sattar to please welcome our first chair with Rose. And yeah, आपस में. So our second chair is here, Dr. Asma Sattar Khan. She is currently serving as research officer in charge in Drug Externalization Research Unit, Ghaziabad. She completed MSc Chemistry in 1993 from Lucknow University. She also got PhD in 2000 from Lucknow University. Before joining CCRM, she worked as lecturer from 1998 to 2006 in Islamic Degree College, Lucknow. She has given prolific contribution in the publication of various Unani pharmacopoeias. So, to welcome our second chair, Dr. Asma Sattar, I will request our first chair to please honor. So thank you very much. As we are already running late, I would hand over the mic to our chairs to please start the proceeding. Thank you very much.
She graduated with a BMS degree from the Institute of Indigenous Medicine, University of Columbia, Sri Lanka, with first class honor in 2005. Afterwards, she completed her MD in Immunology at Hamdard University. Voice is enough. They all of them. Afterwards, she completed her MD in Immunology at Hamdard University, New Delhi in 2012, followed by a PhD in Unani medicine in Unani pharmacology at Jamia Millia Islamia in 2022 and 23. She is the first MD, PhD and professor in Unani Immunology in Sri Lanka. She was also accredited with international accreditation as a teacher in higher education by the Staff and Educational Development Association UK in 2009. Welcome you, ma'am. So I want we welcome you, ma'am, in our Unani Day 2025.
Thank you.
Mic is all yours.
Okay. So, respected chairpersons and distinguished speakers and my dear delegates, Assalamu alaikum. So, I am Professor Hamia from Faculty of Indigenous Medicine, University of Colombo, Sri Lanka. So, this is actually my PhD topic. I did my PhD in Jamia Millia Islamia, part of my PhD. So that is physical chemical characterization and in vitro anti-inflammatory activity of Kushta Godondi, a Unani herbal preparation. So as you know, Kushta is one of the important formulation of Unani system medicine obtained by calcination of metals, minerals and animal origin drug. So according to Unani concept, prepare any drug in the form of Kushta remarkably improves the potency, efficacy and safety of the drug. There are many data available that is available in the market that herbal formulation, it is when we prepare, it is safe when we prepare properly and use it. But okay, so if you prepare in proper method and if you use it, it can cause injuries to the health. So but there are some drawbacks in specialized process of this Kushta related to quality of raw materials and quality of cow dung cake and deviation in the standard manufacturing practices either intentionally or unintentionally, and instability in the intensity of the fire and difficulty in controlling the temperature. Although it is important to validate the traditional claims scientifically in the process of preparation of Kushta, which are responsible for its high efficacy. Therefore, a multidisciplinary approach utilizing modern techniques with classical tests were utilized to evaluate the efficacy and to assess the quality of the Kushta. So, my main objective is to evaluate the efficacy and to establish quality control parameters of Kushta by classical and furnace method. My specific objective are to standardize the temperature required for the preparation of Kushta by classical method in order to adapt it for the purpose of preparation of the Kushta same by furnace method, and also to establish the quality control parameters and to reveal the effect of herb in the preparation of Kushta, and also to evaluate the anti-inflammatory effect of Kushta by classical and the furnace method. So there are many prescription of Kushta Godondi available in the classical text with a different combination of herb using with difficult breakers in order to treat for different conditions. But I have chosen the Kushta Godondi in the National Formula of Unani Medicine Part 1, which contains gypsum 100 grams and Vithanea somnifera decoction 100 ml. So it is one of the important preparation having properties like mucoviasa, mucoviaba and valiliver, used in hemiplegia, facial palsy, numbness, arthralgia, gout and pyrexia and cramps. So this is my material methods. So I have used both this gypsum and the Vithanea somnifera. So I have prepared 3 samples. One sample I have prepared according to classical method. So while doing that, I have recorded the temperature every 10 minutes in order to prepare the thermogram. So the second sample is prepared with the tubular furnace using the modern techniques, and the third sample I have prepared according to classical method. But instead of Joshanda, I have used water to check the effect of the herbs in the Kushta. So after that I have done the standardization according to classical and the modern method, and I did the anti-inflammatory studies that in carrageenan induced for edema model. So I have assessed the for edema and the antioxidant effects and cytokine and the histopathology. So I have collected the raw materials from New Delhi and I have authenticated from NISCA and the Department of Chemistry, Jamia Millia Islamia. So I have prepared the Kushta. Small pieces of Kushta Gondoli were cleaned with the hot water, then I have ground with the decoction of askan. We prepare the cake and we undergo glacial mud process. And we use 25 kg of cow dung cake to prepare the Kushta. So the name, we have termed this name, the preparation as a KGC, that is Kushta Gondoli classical method. So the second preparation I have prepared I used the same classical method, but here instead of Joshanda I have used the water in order to reveal check the effect of the herbs. So third sample I have used the furnace method, the tubular furnace. So I used the modern grinder and the syringe to prepare the tikiyas and the laboratory oven and use this tubular furnace. So this is my thermogram, that is thermocouple I used to record the temperature. So you can see it went up to 950 degrees Celsius. I maintained for 1 hour, then I am allowed for the natural cooling of the drug. So these are the quality control parameters I will discuss in the results and discussion. So anti-inflammatory I use 4 to 6 weeks of all male star rats weighing 130 to 160 gram, and I use the standard housing, environment and food. So I use acetylsalicylic acid as a standard drug, 100 milligram per kg, and high dose of, that is all 3 samples I have used, and low dose of KGC and KGF, that is I use in the, not I didn't use the KGW, and I use 1% of carrageenan as a to induce edema. So I have divided my animals into 7 groups having 6 animals each. So drugs were given orally, that is 1.5 to 2 ml via gauge needle, and after 1 hour the edema was induced with 1% carrageenan. So PA edema was recorded at the interval of 1st, 2nd, 3rd and 5th hour after the injection of carrageenan by prothostometer. So these are the groupings of these my studies. So at the end of this study I have I calculate the inhibition rate using this formula. So after end of the experiment I withdraw the blood through retroorbital plexus. I have centrifuge. We separated the serum and we kept that for in minus 220 degrees Celsius to analyze of MDA, catalyst activity and total antioxidant activity. So tissue was collected of the injected joint and for the histopathological analysis and for pro-inflammatory markers, that is TNF alpha, IL-1 beta and IL-6. So after that we euthanize by decapitation under overdose of anesthetic ether. So determination of MDA level. So maltodextrin is a final product of lipid peroxidation which produces a color complex when it reacts with barbiturate acid reaction. So we use this sample 0.2 ml and as a control we use the distilled water. So we mix with the reaction mixture, you can see, and we incubated for 95 degrees Celsius for 60 minutes. Then from that we have taken the 1 ml of substrate and we add 1.5 ml of 10% trichloroacetic acid. Then we have centrifuge. So after that we have taken the optical density with spectrophotometer using wavelength of 532. So after that the results were applied in this formula in order to determine the MD level. So total catalyst activity was prepared through this. Each sample we have prepared this and the reagent and the H2O2 and the blank. So after that we have taken the spectrometer reading at 410 nanometers. So the results were applied to this formula in order to determine the catalyst activity. So again the total antioxidant activity same as a catalyst activity we did. We used the sample 0.2 ml and preparation of control 0.2 ml redistilled water. We mixed with the reaction mixture and we incubated for 45 hours at 40 degrees Celsius. From that we have taken 2 ml of substrate. We add 1 ml of 40% of trichloroacetic acid. Then we centrifuge and we have taken 1 ml of supernatant. Then we add for the 2 ml of 0.2% of aqueous thiofluoric acid, and we boil for 15 minutes. Then we took the optical density in 532. So again we apply this to the formula. So then we done the analysis of pro-inflammatory markers. First we isolate the RNA, so then we check the quality of the RNA, then we synthesize the cDNA synthesis, then we check the cytokines gene expression by PCR, then gel electrophoresis, then gel imaging. So we analyze with the ImageG software. So these are the gene and their primer sequence we have used to do this analysis of pro-inflammatory markers. So after that we have done the histopathological analysis. So statistical significance of the group were evaluated with mean and standard deviation using the one-way ANOVA followed by Dunant post hoc test. The p-value were looked significant at 0.05 or less. So let's come to the results and discussion. So first my part is quality standards of Kushta. So these are the quality tests, classical test we have done that regarding the color, taste, loss of metallic cluster, fineness test, floating test, grain test, smoking test and wall test, so all positives. So next we did the physicochemical analysis. So you can see there are many parameters like bulk density, tap density, Hosner's ratio, Cas index, pH of 1 and 10% solution, loss and drying, total ash, acid insoluble ash, water soluble ash, methonic extract, aqueous test. So you can see the next chart, that is important because we have so shall I skip?
No, you can take 5
Okay.
minutes.
Okay. So you can see that these are the significant levels. So these are the XRD of this my study. So this is you can see the first one is gypsums, the peak. So it is shifted towards right in the all three Kushtas. So you can see this in the its new formulation has formed. This is gypsums of 2 theta value. These two are the classical and the furnace method. So it is almost equal. So this is the water which contain Kushta. So this is spectroscopic analysis. You can see peak is shift to 228 millimeters. So this towards the high energy level. So FTIR, you can see the separate FTIR of all 4 samples within gypsum. This is the combined sample. You can see water molecule is completely evacuated and the SO bond is prominent. And this is you can see here even in the gypsum, most of the functional group it is removed in the Kushta, so it is purified well. So this is scanning electron microscope results. So this is EDX. So zeta potential. So high zeta potential having good stability, easy dispersion, while substance with low zeta potential is vice versa. But here our zeta potential is very low. So heavy metal analysis you can see there is not detected. So results of anti-inflammatory study, you can see results of PA, edema in each hour. You can see in the brackets I have put the inhibition rate. So this is the graph shows the same, and the next graph is of the percentage of inhibition on 5th hour. So antioxidant activity, again you can see the significant and non-significant levels you can observe. So the graph shows the same. Pro-inflammatory markers, again TNF alpha, IL-1 beta, IL-6. There are non-significant results also in the low dose, but high dose shows very significant results. So this is also the graph shows the same. These are the histopathological analysis. You can see more edema. First side is with gypsum only. So other you can see mild inflammation in this third group tissue with moderate. And actually high dose showed good results than the low dose. My conclusion is Kushta Godandi was prepared by classical method and the thermogram was developed. This can be utilized as a standard reference to prepare the Kushta by furnace method or any other method. So results of standardization of Kushta Godandi showed no any obvious changes in the formulation of Kushta by classical and the furnace method. However, variation detected in the Kushta formulated with or without Ascan. Advanced analytical technique used in this work grant an applicable correlation between the traditional information on characterization and processing of Kushta. The results of physicochemical analysis tests established in the study may be regarded as a standard reference for Kushta Godandi to give safe and effective medication for mankind. So this is my regarding conclusion of my
anti-inflammatory study. Carrageenan-induced inflammation shows suppression of edema and decrease in serum MD, increase of catalase activity and total antioxidant activity in the test group than in the carrageen control group with different significance level. Effect of suppression of release of pro-inflammatory cytokine not evidence in low dose of KGC and KGF and higher dose of KGW when compared with other groups. However, groups animal treated with acetylsalicylic acid in a higher dose of KGC and KGF exhibit significant improvement in all test parameters such as inhibition of poedema, antioxidant activity, suppression of pro-inflammatory markers with different significant levels and also in the histopathological analysis. So thank you very much.
Thank you, Dr. Nazmeen Fahamia. And any question? Any of you want? We will take because we are short of time. Thank you very much, ma'am.
Okay, thank you.
तो उनका वो मोमेंट होते बाद हाँ प्लीज रेडी उनका मोमेंट दे देते शी इज या नो प्रॉब्लम
As Dr. Fahmiya have another program, so I would request our chairs to please honor Dr. Nazim Fahmiya with memento as to token of appreciation.
Please.
Understanding the situation.
No, you are most welcome. And bag is for you. Please.
Okay, thank you very much, sir. Thank you.
He is my senior.
Assalamu alaikum. Our next resource person, Muhammad Ishaq Gir, Professor, Department of Pharmaceutical Sciences, University of Kashmir, Srinagar. He will join us by online. Professor Gir Muhammad Ishaq is a senior faculty member at the Department of the Pharmaceutical Sciences, University of Kashmir, with more than 22 years of teaching and research experiences. Additionally, he has served as a coordinator, UGC Human Resources Development Center, and as a director at Center for the Career Planning and Counseling of the University of Kashmir. He has organized several faculty development programs, placement, drives and coaching programs also. He has delivered 175 invited lectures outside of this university. Muhammad Gir Ishaq, Professor Gir is also member of several state and national level committees, including advisor, price monitoring resource unit member, PG Curriculum Committee, Pharmacy Council of India, member coordinator, NAC peer team, nominee CCSEA, member editorial board of the several high impact national and international journal of repute. Muhammad Ishaq Gir, please start.
Respected chairperson, respected co-chairpersons, ladies and gentlemen, a very good afternoon to all of you. First of all, I would like to thank the respected chairpersons for giving me this opportunity to make. I could not join the conference in person. Therefore, I had made a request for making my presentation online. Thanks for acceding to my request.
1 minute, please.
Allowing me to make this presentation on the topic emerging role and significance of Unani medicine in the world of integrative, holistic and lifestyle medicine. Well, this topic struck my mind as a result of an editorial that I had recently published in a journal, Journal of Integrative Medicine and Public Health, which was entitled Integrative Holistic Medicine.
Please, listen 1 minute, please. Sorry for interruption. Sir, you have only 10 minutes. Please conclude within a 10 minutes, if you don't mind, please.
Okay, sir. Okay, sir. Thank you. So this was the topic of the editorial, because of which I chose the present topic of this presentation. And the whole idea is, there is a lot of talk about integration and innovation in traditional systems of medicine. Well, there is a paradigm shift being witnessed worldwide and enlightening in the transition of healthcare from passive to proactive healthcare, in the transition of healthcare from patient-centered to people-centered, and from disease-centered to health-centered healthcare. The idea is to promote preventive, precision, personalized, and proactive healthcare, to promote self-rule and self-discipline, co-construction, and sharing. Now, in order to save time, I will skip the definitions of integrative medicine, the definition of holistic medicine, and the definition of lifestyle medicine. But as already known to all of us, that there are perils of modern lifestyle, which leads to a host of diseases. And there are also several hazards or perils of modern medicine, like adverse drug reactions, medication errors, polypharmacy, drug interactions, non-compliance, self-medication, substandard and fake medicines, irrational use of medicines. So there is this thinking that there has to be a holistic approach towards healthcare, which takes care of disease prevention, health promotion, primary care, diagnosis, treatment, rehabilitation, and continuing care at the same time. And when we look at the traditional systems of medicine, they are based on certain ideologies, certain concepts and principles. Like in Unani system of medicine, we have the theory of humus put forth by the father of medicine, Hippocrates. And in Ayurveda, we have the principles, the basic forces which govern the physiologies of the individual, the Vata, Pitta, and Kapha. And therefore, on this basis, we divide or categorize patients into several prakritis in case of Ayurveda and into several mizaj or temperament in case of Unani systems of medicine. So the idea here is that we need scientific validation and standardization, and we need to evolve scientific basis of this temperament, as has already been done in Ayurveda by virtue of the Ayur Genomics project, which was launched by the CSIR GIB. And same thing needs to be replicated or emulated in Unani system of medicine. And we need to have some kind of physiological, biochemical, pathological, or genetic basis of this temperament. And we need to establish this by virtue of conducting the scientific studies. We already know that Unani system medicine is per se holistic and integrative medicine. That is, it emphasizes regimenal therapy, dietotherapy, pharmacotherapy, as well as surgery. Similarly, in Ayurveda, there is multimodality treatment being propagated, and which are also the 6 pillars of lifestyle medicine. That is exercise, trust management, social support, sleep, avoiding risky behavior, and nutrition. Same thing is propagated through Unani system of medicine through Asbab-e-Sitta Zaruriya. And then there are 10 parameters, the Ajnas-e-Ashara for determination of temperament. So using this Mizaj stratification, we can evolve or generate some genetic basis by conducting some stratification studies or scientific studies, as has already been done in case of Ayurveda, wherein healthy individuals of contrasting prakritis have been identified, and the gene expression studies have been conducted, genome-wide, phenome-wide studies have been conducted, the biochemical and hematological differences have been observed, the physiological parameters have been recorded, and on that basis, stratification is helping in accelerating the discovery of markers for predictive, preventive, and personalized medicine. Therefore, there are immense benefits of integrative medicine in chronic conditions like arthritis or heart these diseases or blood pressure or stroke, obesity, menstrual disorders, PCOS, infertility, etc. So there is need for innovations needed for integrating Unani with other healthcare systems. There is need for standardization and scientific validation, integration with modern diagnostic tools. A lot of buzz is being heard about the artificial intelligence, the cloud computing, data mining, robotics, blockchain technology. This needs to be integrated into the Unani system of medicine, so that there is interdisciplinary education and training, there is research and development, there is personalized medicine and precision health, we need to have adequate regulatory policy frameworks in place, and we need to create public awareness and advocacy. Then only we can, make progress and keep pace with the changing times and trends. And there are a whole lot of studies that can be conducted in terms of establishing the biochemical basis, the physiological basis, the genetic basis, and pathological basis of temperament or prakriti, and establishing the differences among individuals or patients. Because we know that in Unani, there are temperaments not only for patients, but also for drugs, diets, and environmental factors. we need by taking inspiration from this Ayur Genomics project of the Ayush the ministry, we need to have similar projects wherein genomic studies must be conducted. A lot of progress has been made since 2002 when this Ayur Genomics project was launched by the government of India, and already they have listed it as among 80 success stories. So similar thing needs to be done in Unani system of medicine. Already in Ayurveda, they have correlated HLA types with Prakriti types. They have correlated genome and phenome-wide studies with Prakriti groups. And a lot of studies have been conducted on high altitude adaptation among different Prakriti types and upregulation of inflammatory genes. So, but unfortunately, when I searched the scientific literature, I could find hardly few papers which have done these kinds of studies on Unani medicines. So my conclusion is that in the coming years, Unani medicine is expected to experience a significant rise in acceptance, demand, and marketing. Factors such as increased awareness of natural health, shift towards preventive care, and aging population and advancements in digital healthcare will also contribute to this particular trend. However, for Unani medicine, it will require continued scientific validation, standardization, and strategic marketing. And by leveraging technological advancements, maintaining high standards, and expanding its reach through global markets, Unani medicine has the potential to become a key player in global healthcare. That's all. Many thanks to all of you for giving me this opportunity. Over to you.
Okay, thank you, doctors. Thank you so much. We will take the question later on.
Thank you.
Thank you so much.
Okay, sir.
Now we start with our third speaker, Dr. Khalid Zafar Masoodi. Dr. Khalid. अच्छा आसिया सुल्ताना. Now we start with Dr. Asiya Sultana. Professor Asiya Sultana is a distinguished professor serving as a chairperson in the Department of Ilaj bil Tadbir, Regimental Therapy, Faculty of Unani Medicine, Aligarh Muslim University, Aligarh. Professor Asiya has around 25 years of academic and research experience. She is widely recognized for her contribution toward the promulgation of Unani medicine at national and international levels. Before joining Aligarh Muslim University, she has served Hamdard University, New Delhi as an assistant professor and consultant physician for more than 11 years. So, welcome, ma'am. The stage is all yours. 10 minutes, hardly 15, ma'am. We are short of time.
Honorable Chair of the session, esteemed guests and delegates. First of all, at the outset, I would like to express my gratitude towards the organizers of this program. Indeed, a very important one, the celebration of Unani Day, and to spread word about the wisdom that Unani holds for centuries. I would like to talk about a very burning issue today, and if I think I can make even few of you to think and to revisit your eating habits, I think my job is done. And why I chose this topic, I will try to conclude within time. So I would like to why I chose this.
Very good. You can take 5 minutes extra.
Thank you very much. I cannot come and make an unnecessary delay between you and your lunch. So I would like to conclude this in a very precise time. So why I chose this topic, this particular topic, colonization of Indian food industry, legacy and modern health challenges, is because of the most recent report by the Indian Council for Medical Research, ICMR, that submitted this May only. And it said that Indians are very much prone to metabolic diseases because India is now facing double onslaught. The communicable diseases are still there, and the non-communicable diseases have started taking toll on Indian population. And the reason being is the unhealthy eating habits. And if Indians can curtail on their sugar and their salt intake, that can substantially reduce 60% of the cardiovascular diseases, liver diseases, and metabolic diseases can be reduced just by curtailing on sugar and salt. So this is a very small statement, but it has very larger impact, and this is why I chose this topic. Next, please. It is a time of globalization. It's an era of globalization. It is good that that the entire world has become a small village, but we have to think we can learn many languages, we can imbibe many other traits that are prevailing globally, but when it comes to eat, we need to be very mindful. And this is the main slogan that I have put on my cover page, that think globally but eat indigenous. And this is what the father of medicine, Hippocrates, has said, that the food should have 3 qualities only. Number 1, it has to be local, it has to be seasonal, and it has to be traditional. So these are the contents, the political colonization. India was first colonized. It was a political colonization. It was a political influence that India was under slavery of British rulers. But now this is the time, and fortunately we now gain our freedom. But till this time we are slave in our mind, and we unknowingly or sometimes knowingly we follow the Western tradition. And this time it is the food industry which is getting colonized, and we are acquiring unhealthy and exotic food habits that are very antithetical to health. Then British rule, that the distribution of Indian food system, the modern food industry, and which is antithetical to health, and rise of ultra processed food. The supermarkets and this departmental stores, they have added further to this problem by providing this rigorous marketing gimmicks and selling all those unhealthy stuff, especially the children are target. The psychological and economic slavery of food industry, and how ultra processed food impact health, and what can be the solution and way forward. So India has a rich food heritage with regionally diverse culinary practices. India is a country of pluralism. There is lingual pluralism, there is cultural, medical. In every sector we have pluralistic culture, and this is the beauty of our nation. And India, it enjoys a very rich food culture. Food which is available in south is entirely different from north and west and east. So no any country is having that kind of diversity in food, and we are very much masters. We are learning things from west now. They are becoming simple in their food. So we are learning things from western world, in which we were masters once. Colonization and industrialization transformed the dietary habits grossly. When India was under British rule, it greatly impacted the eating habits, because the Indian officials who used to follow their British superiors, they used to copy you must have heard the dabba food, the dabba meal prevailing in Mumbai. That is also under influence of the British government. So and the impacts are still very long lasting and are still witnessed. But now what we are doing, this modern food and ultra processed food is gaining popularity in culture, in every sector. And food which was the marker of identity of the religious ethnicity, kinship, geographical distribution, everything is now becoming a marker of you can say a marker of contemporary and upscale lifestyle system. So in the name of modernity, we are acquiring things which are very unwise, and we need to do away with this. So this is a brief account of the colonization. The colonization of India by British Empire had profound effect on various aspects of Indian society, including the food industry. And British colonization introduced few food policies and ergo-economic they overpower the ergo-economic and they force the farmers to grow the things that were not available in their country and which help them in revenue generation, just entirely overlooking the need of the Indian population. So the diverse diet, which was the very much part of the staple food of India, like millets, pulses, fresh produce, and medicinal herbs, they were totally overlooked. Sustainable farming was very much kept aside. Then community food system and self-resilient economy, it was all crushed under the British rule. And food as the slogan of food as medicine, which was very much owned to India and Indian subcontinent, it was at stake that time. The economic and agricultural exploitation was also, I'm just skipping this, and would like to share one thing. Many of you must be aware about the famine and starvation of the Bengal that took place in 1943, killed millions of food because food was exported to Britain. And the kind of food they forced the farmers to grow, which it was totally according to their own choice. So there was a paradigm shift from self-sufficiency to cash crop dependency, food cultivation of tea, sugar, coffee, and indigo for exports, and decline of traditional grains like millets and pulses, introduction of refined flour and maida with sugar and polished cereals it took place and it replaced all the indigenous food. And then this is the picture of the Bengal famine that took place in 1943, and it is written as a black spot in the Indian history when it comes to agroeconomics. So the modern food industry, it is now becoming increasingly popular in the name of convenience, in the name of modernity, and the food is also relatively affordable. And according to the busy lifestyle, and also it has got taste enhancers. So, and this supply system like Swiggy and Blinkit and all these, they are creating, multiplying this problem manifolds. So contemporary food industry, heavily processed food, ready to eat, add high sugar, they are replacing the rich, nutritious traditional food with the empty calories. Because what is available in such kind of food, these are the empty calories, and at the same time we are causing obesity, which is the mother of all the metabolic diseases, and at the same time it is also causing nutritional deficiencies because these are the empty calories. Marketing strategies like celebrity endorsing these products, and then they are reinforcing the concept that these fast food are not only very convenient, but they are also the need of the hour and a status symbol. So children and the modern young adults, they are becoming increasingly target of this concept, and we have to free our mindset from this colonization. So nutritional misinformation, labeling techniques, and health washing. Health washing is, telling untruth and telling what is not printed on the label. And economic, emotional appeals and celebrity endorsement, this is all adding to this Indian culture, which is getting entirely, towards devastation. Then introduction of Western food. The psychological and economic slavery of food, once colonized by force and now it is colonized by choice. India, once a food exporter, now depends on the multinational food brands. We are self-sufficient. हम कृषि प्रधान एक राष्ट्र है, लेकिन हम उन लोगों पर अभी भी फिर से दोबारा से उन पर डिपेंड हो गए हैं for our taste buds, for our gastronomic desires. And Western food culture dominates youth preferences and lifestyles. So role of schools and families in changing food habit include Indian superfood in school meals, promote home cooked meals in families, and educate children, and health education campaign in schools. It is very much needed on traditional food wisdom. Of course, there are several government policies like Food and Safety Standard Authority of India that has regulatory guidelines, but we all know that how much implicable they are and how much effectively they are working. The nutrition campaign like Eat Right India, this is the most recent campaign that India has started, Eat Right campaign, and all of the government organizations are supposed to get this certification. Then banning junk food and schools in promoting millet-based diets, and midday meal scheme now include millet. This is the most recent, and we know that 2023 was celebrated as the International Year of Millet Awareness, and now we have just started to revisiting our eating choices. So I would like to share some of the culprits that are responsible for all the modern health challenges. Food like sodium benzoate. आप सबको पता होगा, you must be quite aware about the food, soft drinks, juices, pickles, sauces and salad dressings. The jam that we made, the sauce that we made at home, the expiry is only hardly 4 or 5 days. But what kind of jam it is and what kind of sauce it is that is not rotten even after 2-3 months and without any refrigeration. So anything which is not getting rotten is not healthy for the human body. Then monosodium glutamate is a taste enhancer which is available in many of the food items, and it is having severe carcinogenic properties as well as the cognitive decline, especially for children. Then artificial sweeteners, aspartate and sucrose, these are responsible for insulin resistance, which is very much common among young adults and even in adolescents. High fructose corn syrup, it is being used in so many baked goods and packaged juices, and the mechanism is it metabolized in the liver and converted into fat, leading to fatty liver. Earlier it was alcoholic liver disease, but now it has become non-alcoholic liver disease, and whoever is not consuming alcohol is getting targeted, reason being these kind of food additives, condiments and seasoning, which is being used in so many food items in the name of taste enhancing. And also because of the extra palatability, people are also get into overeating. And then sodium nitrite, phosphoric acid, food in soft drinks, packaged foods, processed food mechanism, disrupt the calcium absorption, leading to decrease in bone mineral density, osteoporosis and increased risk of kidney stones. And then BPA, found in canned food, plastic containers and food packaging, mechanism of action, it acts as an endocrine disruptor, leading to hypothyroidism and PCOD in young girls, leading to infertility later in their lives. And then mimicking estrogen and potentially causing hormonal imbalances, infertility and early puberty. Artificial colors are very carcinogenic, we all are aware about it, causing so many skin allergies, and that is why the skin diseases which are now prevalent was totally unknown in the past times. And of course, the kind of drugs which are available for the treatment they are having inconsistent effects. So they are becoming more and more incurable. And potassium bromate, food, packaged breads, bakery products, flour-based snacks, they consider the carcinogen potassium bromate may increase the risk of thyroid cancer and kidney damage with prolonged consumption. So we are having so many variety of mithaiyan, sweets that are available in India. Every region and every religion having their own kind of sweets and then dishes. And what is the need to which we are not accustomed with that pasta and pizza and burger. These are not the items for our own plate. Like the climatic factor is different, like our epigenetic factors are different, the internal constitution is different. So we need to look for the traditional food, the local food, because it is free from additive. This concept that food has to be local. If it is local, it will be sustainable, it will be fresh, and it will be free from all kind of preservatives. And the bread that comes to our table, when we call it fresh, it is already 6 days old. So it is a time that we need to think the home cooked food, which is the need of the hour. And the traditional food embodies this wisdom, offering balance and well-being in harmony with nature. apart from destroying the individual health, it is also against the environment. It is not a, now the concept of planetary diet is also being introduced, planetary diet which is sustainable and very in harmony with our environment. So supermarkets promote packaging food over fresh home cooked meals. This has led to decline in the consumption of traditional nutrients rich diet, increasing risk of lifestyle diseases, and many people are moving away from nutritious home cooked meals to fast food and Western style diets, leading to nutritional deficiencies. And now the celebrities who are endorsing these kind of food, they are consuming home cooked food in their workplaces. In Unani system of medicine, many modified diets separately or as an adjuvant with pharmacotherapy are also used for treatment of various diseases, and these are the food very much in accordance with the Hippocratic code that let food be your medicine. These diets have been mentioned. Okay. So this is Mauche, you are already aware, barley water, gruel, hydromel. These are the very popular classical food items that are the need of the hour, and that will definitely, the good news is about these problems is they are quite reversible. So one can easily reverse this pathological process by using all these things. And how the ultra processed food impact health, you can see the trans fat and all these things. So savoring tradition, reviving heritage and gains and food in age of the refined foods, say no to refined foods, then colonial influence, we have to get the our mindset change, because progress is equal to the difference between the mind and the mindset. So we have to bridge this gap, and nutritional laws we have to make by conscious efforts and by our mindful choices, and promote healthy eating habits. We can replace soft drinks with buttermilk, sattu, kanji, and also many other regional and traditional drinks are available. And healthier shift, revive traditional wholesome food. So this is my message here, to revisit our food habits and to do away with all the kind of stuff which is available in the name of modernization and convenience. Thank you very much.
Thank you, doctor. Thank you so much.
Now we should think before eating anything, it is healthy or not. Thank you, ma'am. Now, next.
Yes. Dr. Khalid Zafar Masoodi is an associate professor and senior scientist in Division of Plant Biotechnology at Kashmir. He earned his master and doctorate in biotechnology from University of Jammu, later pursued the postdoctoral research at the University of Pittsburgh, USA. He has made significant contribution in oncology, plant biotechnology and molecular biology. He holds 8 patents, including breakthrough in the anti-cancerous therapies and medicinal plant research. Thank you, doctor. Please start.
Thank you, worthy chairman and delegates and organizers. Assalamu alaikum.
Please, doctor, please keep in mind only 15 minutes. 5 minutes is still given to you, only 15 minutes.
Yeah.
You need to conclude within the 15 minutes.
Okay, I'll try to conclude in 15 minutes. Our studies has led to 8 patents that we have gained in the last 10 years of my service at SCOST-K. Before I move on to the research, there are some stats that 1 in 3 people die of coronary heart disease, and there were 20.5 million people died of the coronary heart disease. There was a 500,000-fold increase in diabetes in the last 120 years. If you talk about Alzheimer's, 1 in 3, every 3 seconds we have 1 Alzheimer's patient, and it will be 150 million cases by 2050. So we want to look like this, but actually we are this, and there is a 35-fold increase in obesity in the last 120 years. And talking about childhood obesity, there is a 10-fold increase between age group 5 and 19 in childhood obesities. And talking about cancer, there's a 62-fold increase in cancer in the last 200 years. And we know that cancers, 20% of the cancers are genetic. If someone has a family history of cancers, maybe there's a chance that he'll or she will have the cancer. And 80% are from the food we eat, or the air we breathe, or the radiation we encounter. So I will not repeat what Professor Sultana said. So there's a shift in food habits. The processed foods that we eat around 70 kgs of preservatives every year. That's equivalent to 700 burgers per year. And most of the foods we see, these preservatives are being banned. Like in 2016, Government of India banned potassium bromate that was used as bread softeners. Or like we say, Nestle recalled this ice cream because they found heavy metals in it. Or aspartame, recently WHO said that it causes cancer. So the small toys we give to children when they take a bath, these are like soft toys. So they contain, or the plastic bottles that we use to drink water, they contain phthalates, they are endocrine disruptors. And the makeup that ladies use, they have these parabens that permeate through the skin, and increase chances of breast cancer and reproductive failures. So there are more than 200 types of cancers, and I've been doing drug discovery on prostate cancer. It's the second leading cause of cancer-related deaths in men, and 1 in 7 men are diagnosed with cancer. Besides this, there's a prostate enlargement, that's BPH, or benign prostate hyperplasia. Above the age of 50 years, 35% of the men have BPH, and above the age of 75 years, 50% men have BPH. So these are some of the symptoms associated with enlarged prostate or prostate cancer. You have enlarged prostate, painful urination, incontinence, difficulty in urination, and interrupted urine flow, or you have to wake up at night frequently for urination. So what we did, so this is a small mechanism that I want to show. That the main gene involved in prostate cancer progression is androgen receptor. That's a key gene, and all the therapies in the market, they are against this androgen receptor. So this androgen receptor is a transcription factor, the green portion that you see on the slide. So it is mostly in the cytoplasm. In presence of DHT, it goes in the nucleus. So once in the nucleus, since it's a transcription factor, it puts on all the genes that are involved in cellular proliferation. So, but what happens in castration-resistant prostate cancer, this androgen receptor goes in the nucleus in absence of DHT and stays there. When it stays there, it puts on all the genes that are involved in proliferation, and the cancer grows. So the goal of our study was take this model, this is a C4-, which is a recurrent prostate cancer model. We took this model and we took the androgen receptor gene, we fused it with green fluorescence protein, and then we transferred those in the C4-to cancer cells. Once we did that, you can see that predominantly the androgen receptor is in the nucleus. So our goal was to find the molecule that could abrogate this nuclear localization of AR back into the cytoplasm. So what we did, when I joined SCOS in 2014, so I knew that we have a huge repertoire of medicinal plants in J&K, the Himalayan region. We collected so far more than 400 plants and made a library of around 25,000 extracts. And then we took the cells, we transferred them with GFP fused androgen receptor, and then we screened out these 25,000 extract library. And we found out 16 hits that would abrogate the nuclear localization of AR. We're very excited to get these results. And then we checked the expression of AR or the PSA, that's the main marker in prostate cancer. We found out that it's getting downregulated, this expression. And then what we did a TLC and took out all these spots through column chromatography and checked these for abrogation of androgen receptor, and we found that one of the spots here, it could abrogate the nucleus of AR. We got the structure, and then we eventually did further study, this the wound healing study, in which we see the migration of, on the left hand side, the migration of cancer cells. It prevented, it decreased the proliferation, it decreased the expression of androgen responsive genes, calreticulin, PSA, ELL2, AF2. And then we did a xenograph study in which we took the mice, put the C4-2 cells mixed with materials in the mice in the flanking regions, and then let it grow to 200 millimeter cube, then castrated the mice, and then treated with our this molecule. And we found out that the life expectancy doubled in mice, as well as decreased the tumor volume in mice. And in some of the mice, the tumor almost in fact vanished. So this is the IHC of PSAR. We found that it was downregulated. The cytotoxic, it was not cytotoxic. And then what we did, we took these tumors and did a comparative RNA-seq. So to find the genes that are being involved in this cancer. So, and also the molecule that we found, we checked its docking with the androgen receptor. It docked quite well, had a good score. And then we did comparative RNA-seq and found out the genes that are involved downregulated, and we found many genes that have a role in prostate cancer are downregulated due to the molecule taxo. We named it taxo. So after this, what we did, we went to FSI. Once we had the molecule, in fact we had 16 molecules, new molecule that we discovered, from which we got so many patents, and many are in process. So what we did, we went to FSI and we saw a clause there that there are already edible recommended daily allowance of various plants. We took those plants in our study and we made a nutraceutical we call Magic for Taxo. So we got FSI approval, we got the patent for it also. And this product is now available on Amazon also. And once we're doing so, I'll skip this. We have a huge market for of these anti-prostate cancer drugs, and people are shifting towards Ayurveda, Unani, or medicinal plants as treatment for various diseases. So once we got that, our university adapted the National Innovation Startup Policy, in which the faculty can also have a startup. So I took my 2 students, make them director 2 and director 3 in company, and we had a company called Kashmir Biotech, and we started selling this. Once we started this, 2 companies, one from Malaysia, H&S Biotech, and one from Canada, SBMAX, so multinational companies. They came to our university, they said, okay, we want to test your products. So we sent our products there, they tested it, it was good, they signed a license agreement with us. Now we export to 2 countries. So the research from lab, we took it to market, and now we're exporting the same. So and also recently we got the approval from Health Canada also for this taxo. So this is the MOU signing, and we got various grants also from IIT Kanpur, so heavily funded us. We were in news, and our startup was ranked 3rd in J&K, most promising startups. And this is the funding we got. And we publish our work in like high impact journals like Nature, Oncology, Molecular Cancer Therapeutics, Endocrinology. So I'll skip this. Yeah, that's all from me. Any questions if someone has? Yeah, we have done docking studies. No, actually, no docking facility. Actually, I'm a biotechnologist. I'm not a bioinformatician, but we have collaborations with one unit in Germany. We have collaborations with University of Arkansas. They did our dockings. Yeah.
Have you do things?
Yeah, they do. Yeah, I'm not an expert in docking, but yeah, we do have people.
As a collaboration mode, you have to.
Pardon?
As a collaboration.
Yeah, as in collaboration mode. Yeah.
Compliance.
Exactly.
Okay, right.
Okay, thank you, Dr. for your wonderful presentation. Any other question? Now you too, Dr. Noman.
First, you have any question? No?
Now, this is very nice session. All the presentations are very good. Actually, this session is all about diet and disease related to diet, and we should eat, as ma'am said, whatever available in our local market and according to the season. But in new generation, they start eating all nonsense, junk food, only burger, pizza and chow mein. And because of that, first is obesity, after that BP and sugar and all, very young age, youngster and getting diseases. So we should think twice before eating all these things and preservatives. So this is very good session and very thoughtful. So we should learn from this. Thank you all the speakers. Thank you very much.
Assalamu alaikum warahmatullahi wabarakatuh. सबसे पहले मैं CCRM का शुक्रगुजार हूँ कि उन्होंने मुझे मदऊ किया और सिर्फ इनवाइट ही नहीं किया बल्कि एक चेयरपर्सन बना के एक इज़्ज़त बख्शी। इसके साथ ही जिस तरीके से ये प्रोग्राम कल से और आज तक हुआ है, उनकी पूरी टीम मुबारकबाद की मुस्तहिक है। ऐसा लग रहा है कि यूनानी को वापस री एस्टेब्लिश करके एक प्लेटफॉर्म दे दिया है, नो डाउट। रही चीज़ आज के ये जो प्रोग्राम था ग़िज़ा के ताल्लुक़ से, और शायद इलाज बिल ग़िज़ा के ताल्लुक़ से ज़्यादा इस प्रोग्राम को दिया गया था, वो मकसद था। हमारे जितने भी स्पीकर्स थे, दे हैव कवर्ड ऑल कॉर्नर्स रिगार्डिंग द डाइट्स, ऑल ओपन एंड हिडन कॉर्नर्स। अगर हम तजर्बे पे तजर्बा करना और तजर्बे को ना मानना, जो मेरा एक थोड़ा बहुत एक्सपीरियंस रहा है कि तजर्बे पे तजर्बा बार बार नहीं करना चाहिए, और तजर्बे पे अगर तजर्बा आपने नहीं लिया तो कहीं ना कहीं प्रॉब्लम होती है। अगर हम अपने एंशिएंट फिजिशियन को देखें तो उन्होंने जो असबाब सित्ता ज़रूरिया, उसका जो 15% जो उसमें 15 या 20% उसमें से एक ग़िज़ा है। दे हैव मेंशन 1000 years एगो, लेकिन अब जाके हम उसकी तरफ जाना चाह रहे हैं जब वेस्टर्न मेडिसिन ने ग़िज़ा के ताल्लुक़ से हमको बता दिया। सर ने जिस तरीके से, खालिद साहब ने भी बताया कि नहीं, कैंसर को प्रिवेंशन करने के लिए यू गो थ्रू द इनिशियल कंडीशन डाइट। तो डाइट का ग़िज़ा का एक अहम किरदार है। अगर आप उसको प्रॉपर फॉलो नहीं करते हैं, जिस तरीके से मैडम ने ये भी बताया, लेकिन यहाँ पर जो डाइट का मकसद ये है इलाज बिल ग़िज़ा कि बहुत सारे जैसे मिजाज में ख़राबी हुई, चाहे वो मादे की वजह से हो या सादा हो, तो हम ग़िज़ा से इलाज सबसे पहले जो क्रोनोलॉजी मेरे लिहाज से होना चाहिए, आप सभी लोग लर्नड पर्सनालिटी बिफोर मी, इलाज बिल ग़िज़ा, इलाज बिल तदबीर, इलाज बिल दवा, देन इलाज बिल ग़िज़ा। तो अगर ये कि सबसे पहले कोशिश करें तो इलाज बिल ग़िज़ा पहले से हम, जिस तरीके से मैडम ने बताया कि हम ग़िज़ा इस तरीके से एडवाइस करें। हम कॉलेजेस की ड्यूटी है कि स्कूलों के अंदर PSM डिपार्टमेंट के लोगों को भेजें ताकि बच्चों को अभी से एडवाइस करें कि जैसा मैडम ने जंक फूड से परहेज़ करें। नहीं तो डॉक्टर साहब ने बताया कितना ज़्यादा प्रिजर्वेटिव अननोटिस, अनवांटेड हमारी बॉडी में चले जाते हैं। तो ये हमारी ड्यूटी तो आप लोगों ने याद दिलाई, बहरहाल हमने रियलाइज़ किया। लेकिन सबसे पहले हमको ग़िज़ा से इलाज करने की कोशिश करना चाहिए, उसके बाद फिर इलाज बिल तदबीर। इलाज बिल तदबीर का जो मेरे प्रोफेसर बैठे हुए हैं, प्रोफेसर अनवर सर, शायद जिनकी बदौलत आज मैं इस चेयर पे यहाँ पे बैठा हुआ हूँ, मैं सर का बहुत बहुत शुक्रगुजार हूँ। इलाज बिल तदबीर एक ऐसी चीज़ है जो मेरा एक्सपीरियंस है, मोर देन 30% जो पेन होते हैं may be treated without any medication only by the इलाज बिल तदबीर। तो हम ग़िज़ा के ज़रिए, तदाबीर के ज़रिए, उसके बाद मेडिसिन और रियत तो दूसरी चीज़ है। बहरहाल बहुत अच्छा प्रोग्राम रहा, बहुत अच्छा ऑर्गेनाइज रहा, और मैं आप सब लोगों का तहे दिल से शुक्रगुजार भी हूँ और मुबारकबाद देता हूँ। थैंक यू सो मच।
Thank you very much, sir, for your invaluable suggestions, comments, and inputs. So now it's time to honor our speakers. So first I would request Dr. Asma Sattar Sahiba, chair of this session, to honor Dr. Professor Asiya Sultana, madam. She is my teacher. Please honor with memento as a token of appreciation. Professor Asiya Sultana. Thank you very much, ma'am. Now I would request Professor Abdul Qavi Sahab to please honor Dr. Khalid Zafar Masoodi. Now I will request once again. I will request Professor Abdul Qavi Sahab to please honor our second chair, Dr. Asma Sattar Sahiba.
इससे बेहतर है मेरी जगह आप उनके सर को बुलाएं, वो ज़्यादा बेहतर रहेगा।
मैं अगर आप लोग अगर कोऑर्डिनेटर्स हो मैनेजमेंट को एतराज ना हो तो आई वुड लाइक टू कॉल प्रोफेसर अनवर सर एंड प्रोफेसर डॉक्टर मोइज साहब, ही इज द प्रेसिडेंट ऑफ द नेशनल यूनानी डॉक्टर वेलफेयर एसोसिएशन ऑफ इंडिया। अगर किसी को इतराज ना हो।
Sure. You are welcome, sir. Please.
मोहित भाई प्लीज कम।
Thank you. Thank you.
Thank you very much, sir. So now it's time to conclude the session. I extend my... Sir, once again I will request Dr. Anwar sir to please honor...
एक मिनट थोड़ा सा उल्टा हो गया।
Dr. Asma Sattar Sahiba.
थैंक यू सर। ये
नाम देख के बताया।
आइए दोबारा फोटो आइए। थैंक यू सो मच।
Thank you very much, sir.
Thank you very much.
As we conclude, I extend my sincere thanks to our chairs and our speakers for their insightful and wonderful deliberations. I extend my sincere thanks to all of you who are present here for your patience and for your enthusiastic and active participation. Thank you very much. Now we break for lunch and we will again assemble here at 2 p.m. Thank you very much.
Wait, sir, wait. Wait, sir.
I'm also please, एक बार
Thank you.
seventh session of the day, that is the day 2 of the international conference, with the theme Translational Research in Unani, Traditional System of Medicine. Before we start the session, we would like to request our chairs to take a place on the dais. We'll start with Mr. Rakhimov Farkod Abduraufovich, Head Secretariat of Minister in the Ministry of Health, Uzbekistan. So please come and take the chair on the dais. I would request Dr. Mujib Hussain, Coordinator of Unani Tib, School of Community and Health Sciences, University of Western Cape, South Africa. So please take the chair on the dais. I would request to take the chair on the dais. And Dr. Parvez Khan, Research Officer in Charge, Regional Research Institute of Unani Medicine, Aligarh. Sir, may I request to please take the chair on the dais? Can we have a big round of applause for our chairs? Thank you. Thank you so much. I would like to request our Dr. Mohammad Iqbal to please present a floral welcome to the chair, Mr. Farkod Abduraufovich. Bilal Ahmed, sir. I'm so sorry for the name. And if I have mistaken any name, I'm really very sorry, Abdurauf, sir, also. Can we have a big round of applause for a floral welcome? I would like to request Dr. Yasmin, Research Officer, to please come and present a flower to Dr. Mujib Hussain. Can we have a big round of applause? Now I would like to request Dr. Neha, Research Associate, CCRM Headquarters, to present a floral welcome to Dr. Parvez Khan. Can we have a big round of applause? Thank you. Thank you so much. Now, we are actually lagging behind time, so we will start the session with no delay. So I will hand over the session to the chairs. Thank you. Please take over, Dr. Mujib Hussain. Thank you.
Okay, good afternoon, everyone. A warm welcome to the session. We'll get straight into it. For our first speaker, we have Professor Farhan Jalis Ahmed, who is the Professor of the Department of Pharmaceutics, School of Pharmaceutical Education and Research, Jamia Hamdan, New Delhi. And we ask him to address the audience, please.
Thank you so much. Thank you so much. Very good afternoon to all the speakers and audience, all the students, respective teachers. I think this is one of the most difficult session. Immediately after a wonderful lunch, I think everyone is feeling sleepy. So my presentation is more about new drug delivery systems. The purpose of this presentation is to introduce all the students about the systems which we are using in allopathy in our drug delivery formulations, because what formulation we are using in Unani system of medicine, they are now modernized in terms of their form, in terms of their appearance, in terms of their patient compliance and availability. Because in modern drugs, we have huge variety of drug delivery platforms, which can also be utilized by in Unani system of medicine, and we can further improve the efficacy, bioavailability, patient compliance, sometimes which is poor when we are talking about sugar-based Unani formulations. Nowadays, those are also transformed into tablets, capsules. Many companies are working onto it, but there are furthermore formulation platforms which can be used for this purpose. So when we are talking about innovation, it can be a simple innovation like which we use in day-to-day life. It is used by in our rural India also, which is known as Jugaad. They have modified many applications, and we have used various types of innovations which are available. If you see in the picture, they are very funny examples, but they are very effective. And similar innovations we can also use. You can see These are the innovations which are used in the rural India, and they use various replacement, various attachments. They have used some vehicles, they have used some canopies, but they are effective, and they are using in day-to-day life to improve their quality of life, to improve their function, all those things. You can see these are, so it is a necessity. You can see this is also a device which is developed by rural India, where they can carry big amount of load, and the load on his neck is much reduced. So to reduce the load on further on all the beams, I think we can use other platforms which are available in systems. Like in modified systems, in our modern system, we use buccal route, nasal route, rectal route, transdermal, paroral, injectables, transdermal, ocular, and then intrauterine, and other injectables like intramuscular and subcutaneous. So these are all the routes which are explored by modern medicine, although these are not routes which are explored by Unani systems. Most of the, I think more than 90% formulations are oral, or paroral, or maybe dermal, topical, sometimes some inhalations are there, insufflations are there, powders are there, but all the routes are not being used. So one message is that I think we should also explore new products by using the old wine in the new bottle. So we can use all those knowledge which is present in our literature, and we can use this knowledge where we can use these routes. Like nowadays, I've seen many companies, they are coming out with nasal drops. Although inhalations and suffocations, they were already there in old literature, but now nasal drops are coming in various forms, like spray, inhalations. So those things are also coming. We can modify our products and to increase the efficacy and efficiency. Similarly, we can have buccal, sublingual, rectal. These are the routes which are high vasculature. The blood supply is very high, so they can be used for immediate action, be in terms of pain relief, be in terms of migraine, or any other relief. That as we are using for angina, we are using sublingual and buccal route for various anti-anginal drugs in modern system. Now drug delivery platform, it has to be whatever platform we are using. In Unani system, mostly we have gums, we have sugar, various types of, these are the main platforms. And the purpose of using this platform by ancient system was to use them as a preservative. So when we are using as a sugar or as a gum, they are not only acting as a preservative, but they are also acting as a absorption enhancer. These platforms, I think their knowledge and their work was experience was so rich that they have understood all these things. Now the advantages are, you have to have very simple, which can be production friendly, because it has to be transported into industry, so it can be used by industry. It has to be in unit dose, so it can be pre-measured. The patient need not to measure it, because sometimes measurement is the aspect which vary from patient to patient. If you say 1 spoon, sometimes he will take half a spoon, 3/4 of a spoon, sometimes he take more than that. So when we are talking about potent medicines, in that case, dose is a very critical. Then it has to be easy to adapt. It should simplify the regulatory process. The process should be easy to acceptable by the regulatory bodies. Then it should used by the large population. You can see this is some more examples. I think top slide it is not visible. It is about osmotic systems. That hiding is not visible. It is about the osmotic drug delivery system, where you can use various types of salts. There are salts like sodium chloride, ammonium chloride. They are filled in the tablet, and tablet is coated with a semi-permeable membrane. And when tablet absorb the fluid inside the stomach or inside the GIT, the hypersonic solid like sodium chloride, it dissolves and create a osmotic pressure. Because whenever there is a permeation through a membrane, its permeation always moves from higher concentration to lower concentration. So it creates a higher concentration inside the tablet, and then drug keep on releasing with a slow release for about 6 to 8 hours within our GIT. That is one model which can be used where we can have prolonged effect of a medicament inside our body. Because a tablet can reside inside the GIT for about 6 to 7 hours, depending upon the disease condition, depending upon person to person variation. It can be up to 36 hours also if he is suffering from constipation or something like that. But it can be easily stay about 6 to 8 hours or 12 hours inside the GIT. So for 12 hours, it keep on releasing the medicine inside the GIT, you will get the uniform effect. This is one example, glyphosate, which is and used in diabetes. It's a very potent sulfonylurea, and it is, if you see the evaluation, if you see this graph, on the right hand side, this is the fluctuation when we are taking multiple doses within the day. You will get fluctuation in the blood concentration. But when you take a single tablet with osmotic control, you get a uniform blood level of the glyphosate. So some more potent medicaments which you are using in Unani, they can be converted into this system. You can see this is optimized tablet. It has very good control. This is the example. Then these are the conjugated polymers. They are used to increase the stability of various medicaments. This conjugation can be used to increase the absorption. We can conjugate our drug, our medicament with a polymer, so that polymer can protect the drug, because our stomach contains acid, small intestine contains various peptide or protein dissolving enzymes. So those protection is not there when we are taking a simple form. Many medicament can be digested, can be inactivated inside our GIT, but we are using a polymer to protect it, to further increase the absorption. If we are increasing the absorption, we can also decrease the dose. Maybe if we are giving 5 grams of a preparation, maybe if we do some clinical study, we can further reduce from 5 grams to 2 grams or 1 gram, depending upon if we can protect it by using these conjugated polymers. This is another example of a control or programmable type of drug delivery system, where you can see you have a capsule which is programmed. You have 3 portions of the capsule. First portion is immediate release, second portion is controlled release, and third portion is prolonged release. So the same capsule can release the drug for a prolonged period of time. So you don't have to take the capsule 3 times a day, 4 times a day. So if the patient is forgetful, suppose you are giving a medicament for Alzheimer, or you are giving the medicament for dementia, but patients are forgetful, they forgot to take that medicament. But if you are giving a tablet once a day, maybe it can be given in the morning, and throughout the day his disease can be controlled. Either it is temperature, or it is blood glucose, or it is a medicament for dementia, all those things can be utilized. You can see this is another platform technology where you have a triple layer in the tablet. The 2 outer layers are of a drug, and middle layer of a second drug, because sometimes when you are mixing more than 2 drugs or 3 drugs, they can be incompatible, they can react with each other. Or if you want to coat a tablet, it can be central tablet can be your main drug, and outer core can be a coating. So protected against acid of stomach, and it will be dissolved in the small intestine where you have alkaline pH, and because of that your stomach is protected, your drug is also protected, and drug is absorbed from small intestine, which you have largest surface area where absorption can take place. This is the example how it is going to be. It can be sustained release where the release is prolonged. It can be pulsatile release where release is after 2 hour, 4 hour, 6 hour, 8 hour in pulses. You can have modified release where release can be prolonged only. You can have chrono release where release is timed. Suppose a medicament for hypertension, mostly in the morning your blood pressure, there are more chances of increase in blood pressure. So it is chronomodulated, so drug will only be released in the morning. It will not release in afternoon or in the evening. In the morning it will release, and it is chrono controlled. This is the few examples of multipolar technology. In this, this is also a very good platform for Unani formulation, because in this case we can give a large amount of dose. You can make your product in the form of multiple small particles, and these particles can be coated with various available polymers, and these polymers are not synthetic. They can be natural, they can be semi-synthetic, they can be biodegradable, but the advantage is your GIT is also protected if it is very harsh, or if it is very sensitive, you can protect your medicament against acid and alkaline media of your GIT. You can also use it for controlling the release of the drug through those small beads. So you can use multiple beads in about 4 to 6 grams of dose, and can be taken by the patient for more effective therapy. This is another targeted delivery in which we attach our drug with vitamin B12. In our GIT we have specialized carriers which can transport the drug from lumen area to serosal area. From inside the GIT, it goes to the blood through B12 carriers. These carriers are basically for absorbing B12 in the body. But we are utilizing this, our natural system. We can coat the drug with vitamin B12. So apart from absorbing vitamin B12, it can also absorb the drug which you want to get absorbed from GIT, especially those drugs which are poorly absorbed.
Sorry, Prof. Ahmed, you have 2 minutes.
Okay. So I will just, I think, hasten to my system. These are all the system which are cochleates. These cochleates are phospholipids, very commonly used nowadays in terms of phospholipid complex with herbs, because they can stabilize and increase the release. These are another example of target system. You can see these are all targeted systems. Micropump. Micropump is used for insulin delivery, where you can deliver the drug. You have a sugar sensor in that pump, and the moment your glucose level rises, it will it automatically release the insulin into your bloodstream. So patient doesn't have to do anything. It will just charge the pump and attach with its body, and it will keep on releasing the insulin whenever it is required. This is pulmonary market, where you can have inhalations, insufflations, all these pulmospheres which can be inhaled by patient for control release. You have transdermal system, which can be applied on the skin. Through skin you can get the drug absorbed, like creams and emulsions and lotions. These are microneedles which can be used for vaccine delivery or delivery of drugs, where we have very, very nano-sized needles which are not felt by human body, but they pierce into the stratum corneum, and then they can deliver the drug. These are all examples of micropore. Then you have Dermastick, which is developed by a German company, where the drug is dissolved in wax. The purpose of this system is to increase the absorption, to increase the solubility. This is all other example. I think I won't be able to complete all the things. I will just needle-free injection technology, which is very commonly used in insulin pumps. You have insulin pumps where air is used to inject the insulin inside the body, because many people, many patients, they have a psychological fear of using the needles. So these pumps are very popular. Now we have inhalable insulin also in the market, although it is not very successful, but still trials are going on. Then you have mucocutaneous absorption, nasal delivery. They all can be used. Ophthalmic delivery for ophthalmic cavity. Then you have various types of liposomes, which can avoid phagocytosis and can stay inside the system for a long time. Then you have vitamin-mediated systems. You have tocosol. There are all the other examples. I think I will not go in detail. You have short of time. So this is the challenges which you are facing for our herbal components which you are using in Unani system. The uniformity of quality control. We are changing the source. We are changing the season. The content of active ingredients changes. The time of cultivation, the time of collection, the time of drying, how it is dried, how it is stored. All those things are affecting the quality. So by using various platform, our purpose is to improve the solubility, is to improve the absorption and viability inside the human body, to prevent the degradation from the biological system, to have effective concentration in the desired area of effect, to have more patient compliance, less cost, and to have better patent life extension. So these are the examples. I think this is one more example of we can make various oils in the form of emulsion which can be applied locally. So these emulsions, they can be made into microemulsions, and these microemulsions can make your oil more acceptable because it is less oily, can spread easily on the skin, and it can also absorb much faster because you are using surface active agents to reduce the surface tension. We can use ternary phase diagram to design this formulation, but this is also very, very good for oil types of products. And we are using these techniques to measure the particle size, because we are making microparticles, nanoparticles, submicronic particles. So all those can be characterized by using these techniques. This is example which we have done in our university. We have converted saffronal aldehyde extract from saffron, and it has very good anti-epileptic potential. So it can be given by nasal route. So we have developed nasal emulsion in the form of drops, like in Unani system or Ayurveda also we take smoke. Smoke is what is colloidal dispersion. So instead of smoke we can take drops which are much easier to take, and these drops have shown to be very effective in absorption in the brain through nasal route. And this is the image of the particles which we have developed. This is, you can see the blood to brain ratio. More it is going into the brain when it is given by nasal route, and this is the effectiveness. This is also one more new formulation which we have developed in the form of bioadhesive vaginal gel. Because of being a bioadhesive, it can stay inside the vaginal cavity. The main problem with existing system is that it is a lot of leakage, staining of clothes. So women compliance is very poor. So, but this system can stay for about 6 to 8 hours, no leakage, no staining of the clothes, and more effective. We have done a small clinical trial in our Majidia hospital, and it is shown to be very effective. These are a few examples. Thank you so much. This is the last slide, the products which we have developed and transferred to industry, some of the papers and publications. Thank you so much.
Thank you so much, Prof. Ahmed. It was a very enlightening talk on innovation and new technology in pharmacy. Thank you. And I will hand over to the chair, Dr. Parvez Khan, to give a comment.
Thank you very much, Dr. Jalis and Dr. Mujib. So very informative details by the Dr. Jalis. I think so the tablet forms and capsules or either local applicants you described very informative for the patient as well as the physicians for the use. Some of the medicine like a spray and the nasal drops is the beneficial. We have our council already used as a arc and the oils in this type of medicines in our practice. So it is a very informative lecture for the patients or as well as the clinicians as well as the market basis. So thank you very much, Dr. Farhan Jalis.
Thank you so much.
I now welcome Dr. Raisuddin, who is the senior lecturer at the Institute of Indigenous Medicine, University of Colombo, Sri Lanka. His topic title is Case Studies. Case studies on ESRD.
Case studies of ESRD, CKD with Unani and
conventional
medicines. Okay. The print is by mistake at the souvenir.
Assalamu alaikum wa rahmatullahi wa barakatuhu. Dear dignitaries and my colleagues, it's very difficult to do a clinical study on ESRD patient or CKD patients. You know, when a person or sick end-stage renal disease person or a CKD patient go to a doctor in Sri Lanka, they will first question is have you ever used Unani or Ayurvedic medicines? Because all these modern people believe Unani and Ayurvedic herbal medicines are causing renal damage. So after that, no patient would like to go to a herbalist, Ayurvedic or Unani physician to find a solution for CKD or ESR. So I got to a few cases in my life since 2014. Alhamdulillah, all cases are living up to date. Nobody died. I got them at almost at the end-stage renal level, and I'm maintaining all of them with conventional and Unani medicines. All are in good health. So please, next slide. Yeah, I have, right. Okay. This is I'm directly coming to the topic because time is very short. This is my first case. The first case came with serum creatinine level 3.8, and his doctor has proposed him within 6 to 9 months he has undergo to dialysis. So he came and asked me, sir, can you help me to improve my creatinine levels and kidney function? I said, of course. And the patient was, you will not believe, 130 kilo. Huge man. So I gave him medicines, and slowly his serum creatinine has fallen down. And up to 2020 March, he was very regular in medicine, and because of COVID-19, so in 2020 March lockdown started, he could not get the medicine. So when he finished without medicine, COVID, you see the serum creatinine level started from 220 to 68. Again, it has fallen down to 3. Serum creatinine has increased up to 3.6. So which button I should click? All right, you see now this is the simple diagram. So the vertical columns are that from serum creatinine is coming down, And when there was no medicine, modern medicine the patient was using, no more Unani medicine, again serum creatinine levels goes up and GFR, estimated GFR comes down. Till now the patient is alive without dialysis or any transplant. But the doctor, consultant nephrologist suggested him in 2014 he should go for dialysis within 9 to 6 months, 6 to 9 months. Okay, this is case number 1. Next. Case number 2 is very interesting. This is a 20-year-old boy and diagnosed with renal failure and uncontrolled malignant hypertension. So the doctor has diagnosed that this serum kidney damage has occurred due to this malignant hypertension. So they were trying with modern medicine for 1 month and no change in the creatinine levels. So many readings within 1 month. Sorry, this case no, go the next, please. Okay, I will present this and I will go to the next. Okay, this case is my own brother. He came with serum creatinine 4.31. In the yellow line, here only we started Unani medicine. Before that, he was under modern medicine, only modern medicine. So serum creatinine fluctuate, it went up to 5 to 6 level, then it is coming to 3.5 and 4.31 serum creatinine. He consulted me, please help me to control this serum creatinine. So from this point, he started medicine and till now he is using this medicine. Yesterday he took his serum creatinine. It was, next please. Yesterday's his serum creatinine was 1.69 with conventional medicine, that is he is taking medicine for dyslipidemia and hypertension and diabetes mellitus. So he is also, which button I should? Which one? This one? Show me. Okay, again the case number, case number 3. Okay, now this is the case of a 20 years old and he came with serum creatinine levels of, for 1 month reading, always ranging about 5.48 and 5.6 and 5.39. So 1 month treatment, modern people could not control the serum creatinine level. 2 nephrologists suggested immediate dialysis and the transplantation. So the father came to me and I started treating him. I started the medicine when the serum creatinine was in 5.82. That is in March 2023. Then it's falling down. And the father has a doubt whether my medicines are reducing the improvement of creatinine levels. So they stopped for some month, 4.5 months, they stopped the medicine because of the fear. Maybe our medicines are reducing the improvement of the kidney function. So after 4 months, the father again contacted me, sir, creatinine level is increasing. Okay, why did you stop medicine? So because of the fear that maybe your medicine is delaying my son's kidneys improvement. Again, they started this and then again see another 15 days level, again creatinine increases. Alhamdulillah, now it is 3.5. Now, until now, no dialysis were done and no other complication, uremic symptom, high fluid level complication, nothing is there. Patient is stable. I hope so, inshallah, he will recover. Inshallah. But up to some extent, not up to the normal level, maybe. Allahu Akbar. This is the style. What has happened? When the gap was there, serum creatinine was increasing. And the case number 4, it's very different case because this patient never takes medicine regularly. Other 3 are taking normally, regularly, but this patient is very poor. Ultimately, only I asked, why do you discontinue medicine? He said, financial crisis. So we have made some arrangement now for the financial crisis. Because I want to help him and I want to know our Unani's findings. So first of all, before starting commencement of our Unani medicine, the patient had serum creatinine 5.38 up to 3.6. Then in July 2023, there was acute kidney injury on CKD. So immediately there were 4 dialysis in hospital within 1 week of time. So ultimately the patient was diagnosed with serum creatinine 4.3. Again, that creatinine level goes up to 8 and then 10. So the patient contacted me while the serum creatinine was 10.47. Very critical case. But he did not develop any uremic symptom or fluid load. Patient was stable, but serum creatinine was all our hospital reports. Very high reports. So we started in this yellow line, July. You will see after 1 month of time, the patient has got up to, alhamdulillah, serum creatinine 2.9. So you see this chart. When he stops medicine, serum creatinine increases, and when he takes medicine, serum creatinine also reduces. I was lucky sometimes because I think, okay, if this is not there, how can I prove the efficacy of our medicine? When they stop medicine, serum creatinine goes up, and they take medicine, serum creatinine comes up. Though it's very clinical, clear evidence that our medicines have some role in the management of serum creatinine level. Okay. So this is the pattern. Now he is now on this stage. He was like this before modern medicine. He got twice as crisis. But all my patients who were administered our medicine, they did not develop any crisis. Alhamdulillah, they are just running their smooth life. But another adding information, in some patients, I added one more medicine, serum creatinine is increasing. I will release at last. Now your question is, what was the medicine, what I was giving? Nothing. I was giving Majun Dabidul and Majun Falasifa and Jawarize Zaruni, half to 5 gram, only at morning, Kali Pete, only empty stomach. Nothing, no special medicine I was administrating. Majun Dabidul Majun Falasifa and Jawarize Zaruni. 3 medicines, a half teaspoon, that is 5 gram, only at morning. Many of the patients I am handling, mashallah, very good results are there. These are the typical case I have ever treated for long time. 15 years, 10 years, 5 years like that. No more crisis. So actually I am a clinician. I have no fund for do very advanced research. So what is the possible mode of action? Why creatinine was controlled? So there may be a creatinine production might have been controlled, or creatinine excretion might have been managed, or both might have been done. That should be done by very advanced research with what is happening when these medicines are given to kidney and what is happening to creatinine levels is a big research. As a clinician, I can propose these might be the function that have been happened through our medicines. So, discussion in combination of Majun Dabidul and Jawariz Zaruni and Majun Falasifa could be more explored as an adjuvant therapy in chronic CKD and in acute CKD, chronic acute kidney injury on CKD. Because if you notice certain cases, some modern people might claim this high range of serum creatinines might be due to some acute chronic injury on chronic renal failure, chronic kidney disease. So we can separate the patients who are having regular creatinine levels, or some are who develop serum creatinine very high all of a sudden. So we can separate the patients and we can try our formula in a very advanced manner to manage CKD. My suggestion is, if somebody else is hypertensive or somebody else is diabetic and they are on modern medication, start these medicines in advance. Because my own brother came with, when he came to me, his serum creatinine was 15. Now he is living around GFR. So GFR, serum creatinine is 4.3, GFR is 15. Now he is ranging in between 40 to 50. So I have, with our medicine, it has been 3 times increased, that GFR levels. Estimated GFR levels have increased 3 times. So if somebody else is anticipated or suspected that they may develop CKD on long-term medication and these type of diseases, we may start in our clinic level to see what's happened. Then we can go for a full-fledged study, because everyone is talking about integration. How to integrate. Without combination therapy, you can't move forward to integration.
Dr. Raisuddin, 2 minutes.
No, I have finished.
Okay. You can round up.
Because I can't steal your time. Thank you very much.
Jazakallah, thank you so much.
Any question? Anyone ask you question?
Yes, please.
Sir, have you ever query for the patient that he has taken some other medicine when he stopped your medicine?
Yes, all are taking modern medicine because people have iman dar hai. Because modern medicine they are believing. For our medicine only they are callous. So they are continuing, they are regularly going. They have their clinic book with me. They are going to the hospital, seeing the consultation. They are very regular in modern medicine. Only in our medicine they try. Well, let me see where I will stop.
One question asked
Yes,
you.
of course.
You use the Majun Tabidul word and those and Madun
Falasifa, Zaruni.
Those are these medicine you have own property or purchase your market?
No market. Very frankly, I use Hamdard medicines.
Oh.
Very frankly.
Thank you.
Very frankly.
Thank you.
Thank you very much.
Thank you, sir.
So this is a very good informative and practice lecture by the Dr. Raisuddin. Thank you very much.
Please take my mobile because I'm sir, 1 minute.
Okay.
I have 1 minute.
Yes, continue.
I am very successfully treating, alhamdulillah, psoriasis, rheumatoid arthritis, and ulcerative colitis, Crohn's disease, and all type of autoimmune diseases. Complete recovery is there. No more complaint, no more medicine.
All medicine used by Hamdard?
1 or 2 local Sri Lankan medicine, baki from Hamdard.
Hamdard.
Patients are no more rheumatoid, no more psoriatic, no more ulcerative colitis. They are free from the disease and free from medicines and free from complaints. Okay. So if anyone wants some suggestion or help,
Thank you very much, doctor. Thank you very much.
Thank you very much.
Thank you very much, Dr. Raisuddin, for that enlightening talk. I would like to welcome now Senior Professor Ahmed Ali from the Department of Life Sciences, University of Mumbai. He will be discussing molecular insights into the anti-inflammatory properties of Unani formulations. Please, sir.
Assalamu alaikum. Very good afternoon. And, respected dignitaries and all the professors and researchers here. Great to see the presentation from all participants. Okay. So without taking much time, I'll go to my topic with the permission from the chair.
Yes, continue.
So, okay. So we have used Unani formulations and we are characterizing the formulation for the anti-inflammatory properties of this formulation, which is a CCRUM grant to us. So I'll not go into the detail of arthritis and mechanism because it's a well-known fact and people have been suffering from this disease since long time, and there are medications available also, but they have their own advantages and disadvantages, and India has a huge burden of this, in fact more than the world average of 0.8% population of the arthritis, and it's increasing because of the lifestyle disorders also. So this is a formulation and Unani conference, so we have been talking about the advantage of medicines, single drug and formulations. So again, I'll not repeat those words. So yeah, so we have used two formulations, Unim H and Unim R, provided by CCRUM with the collaboration from regional center in Mumbai. And we have characterized the, we have done the identification of the plants from Aligarh. So this is the line of the study which we have done for the validation and characterization. So this is a very basic information about the antioxidant, TPC, TFC and all those parameters. So we found that most of the plants have antioxidant activities and they are rich in total phenolics and total phenolides. And we have shown also this by doing the DNA prevention of DNA nicking, DNA damage study. So it can be seen here that we have used PBR 322 in our system, which is a very good system if there is a damage induced to this. The form of PBR changes. So that's a very confirmed study. So we have induced damage with the H2O2, and we have shown that different plants have different potential. So most of the plants could prevent the oxidative stress induced damage to the DNA, and we have identified here some of the biomarkers. We have selected these biomarkers as per the literature available from these plants. They have also very good antioxidant and anti-inflammatory activities. So as per the guidelines and the proposal, we did the acute and chronic toxicity studies. So you can see that we have used two formulations, Unim-R and Unim-H. Unim-R is for dermal application, and Unim-H is oral formulation. So Unim-R, when we did the dermal application, we could show that there is no damage to the skin effects on the rats. So then we did the Unim-H formulation study, and we could find that there is no change in the body weight in grams. And then we did the histopathological study of the vital organs like liver and brain, and from the female and male rats. So we could show that there is no, as such, there is no significant damage to these livers. Similarly, for spleen and heart, we could show that there is not much damage to these organs, as well as kidneys. So we can say that these formulations meet the safety guidelines as per the OECD guidelines. Then we did the chronic toxicity study for 28 days, and then we could show that there is no change in the body weight of this, and there was no motor or behavioral changes observed in the rats. So again, proving the safety of this. And except spleen and kidney, there was no change in the weight of these organs with these formulations. So then we did rat paw edema study using carrageenan, and we could show that there is an increase in, if you can see with the drug that disease condition, there is an increase, but with the increased amount of the formulation, there is reversal of that inflammation. So then we did cotton pellet granular study at one of the collaborative institutes. So we could show here also that with increased 400 mg per kg of Unim-H, there is a complete reversal of the inflammation. Then we did Friends adjuvant induced arthritis study, and here also we could show that at higher concentration of Unim-H or higher amount of Unim-H and Unim-H, that is 50 mg and 400 mg and 100 mg, there is these formulations are very effective. So with this body weight, there is not much change. So then we did the photographic representation of, you can see that with the different drugs, with different concentration of the Unim-H and Unim-R, and you can see that X-rays of this. So you can see here that in the disease condition, you can see that figure 2, there is a swelling of the soft tissues and evidence of the ankle joint analysis, and you can see at higher concentration, higher dose of this Unim-H, that is 400 mg, there was no minimum soft tissue swelling compared to the disease control, and bone architecture was largely preserved. Then we did the cytokine profiling. We have used anti-inflammatory, and we have measured anti-inflammatory and pro-inflammatory. So we could show that there is a suppression of the pro-inflammatory cytokines, that is TNF-alpha, IL-1 beta, IL-2 and IFN, and there was an increase in the anti-inflammatory, that is IL-10, this diagram. So coming to the conclusion, with the time provided, we could show that these formulations meet the safety guidelines, and biochemical or other parameters are also proving the efficacy of these formulations for as anti-inflammatory, so they can be used for, and we are doing some more characterizations like Western blot and other analysis to further characterize and establish that these formulations are very well suitable for arthritis treatment. So thank you. I acknowledge the contribution of my JRFs and my collaborators from Mumbai, and the funding from CCREM and the infrastructure from DST PERS, DST FACE, DST PERS and UGC. So thank you for giving me the opportunity.
Thank you, Professor Ali, for sticking within the time.
Yeah,
thank you so much. I'll pass over to Dr. Khan for comments.
Thank you, Dr. Thank you, Dr. Ahmad Ali. One question ask you, this is the Unim-R and Unim-H.
Yes, sir.
What is the preparation of this one? Either this is...
we have the plants, and we have mixed them in different...
this in the either form tablet, local applicant and capsule?
We have used the Unim-H is oral.
Oral,
aqueous solution, and Unim-R is...
now you explain the 50 and 100 and 400 mg.
Yes, sir.
So which is applied as like 400 and 150 mg?
50 Unim-H.
No, Unim-H.
Yes, sir.
No, 50 mg, 100 mg and 400 mg.
Yes, sir.
किस पर अप्लाई किया आपने? किस तरह नहीं डोज है तो इसमें से क्या रिस्पांस निकला डोज का, जो 3 डोज आपने डिवाइडेड किए?
So the anti-inflammatory property is dose dependent. Yes, at 400 mg there was a very good effect.
Yes. Any question? Anybody else?
Yeah, with the permission of Chair.
Yes. Okay.
Sir, very nice study actually. Sir, have you been informed about the clinical dose? What is the human dose, as sir is asking? Have you been informed that it is?
No, we will be going to that stage after doing this pre-clinical
So it is new
study.
formulation.
Yeah.
Formulation is
pre-clinical study,
yeah.
no?
Yes, sir. It's a pre-clinical.
So have you done any dose ranging study? Because we are also doing 400 mg in animal is very low comparatively. And one more thing I would like to say, sir, when we are doing OECD 407, starting body weight is very high in your study. It was more than 250 mg, 250 grams of rat.
Yes.
It is recommended that you have to take as early as possible, hardly 6 weeks. So 6 week body weight are 140 to 160 gram max. So that is not guideline compliant, because if any changes you are expecting, that should be started as early as possible as per OECD 407. So you should take body weight in future studies somewhere around 150 gram plus minus 10 gram. Otherwise you will not be able to see any changes, because already animal achieved adulthood and there are minimal chances of capturing the toxicological profile of the formulation. Otherwise it is a nice study, sir.
Thank you so much. Thank you, sir.
Yeah, with the permission of chairman, I would like to ask one short question here.
Yes, continue.
Yeah, you use the formulation. Is it a coded one formulation? Formulation. All are the plant ingredient.
Yes, sir.
So, so have you identified the plant, all the plants, authentication certificate?
Yes, sir. We have done it. That was in one slide I showed it actually.
So where you identified that plant?
Aligarh. Aligarh.
So, I would like to suggest you that NISCARE and this FRLST or other people are doing work, and don't take, I think, it's a suggestion only, that don't take the certificate from the side lab people of pharmacy or pharmacology people, because they are not authorized to issue the certificate. Because if you are going for international publication, there is a certain set of protocol, and as per that protocol, the citation and the author name, including the citation, should be proper. And if you go to the guideline of the US FDA or the in NMPB or WHO, you can see that the voucher specimen submission is very much important, and this registration number is very much important thing.
Thank
so,
you, sir. Yes,
yeah,
we
so
have
this
done
is my
it
humble suggestion that try to, because if you are doing some work and your base is not very much clear, so early in the morning in this hall, actually Idris Saab has presented one paper on the DNA barcode itself for authentication and identification of the drug. So that's a major issue. The plants look like same, but they might be different at the variety level, at species level. So that's a humble suggestion for all these preclinical people to do such type of activity before doing for a
We have
large.
sent the plant to one botanist.
Yeah, but try to, yeah, it's only a suggestion for all of you. Thank
No,
you.
very
so
nice.
much.
Thank you,
Thank you,
doctor.
sir.
Thank
Thank
you.
you so much.
Thank you very much.
Thank you. And it's my pleasure now to welcome Professor Shama Parveen. She's from the Center for Interdisciplinary Research in Basic Sciences, Jamia Millia Islamia, here in New Delhi. And her discussion will be on the evaluation of the antiviral activities of Unani drugs against dengue virus.
She is not available. Yes. So, thank you.
Assalamu alaikum and a very good afternoon to all of you, esteemed chairpersons, ladies and gentlemen. First of all, I would like to express my gratitude towards the organizers for giving me opportunity to present our work at this international platform, and I congratulate the entire organizing team of CCRUM for doing such a wonderful job. And they are regularly organizing this Unani Day, and yesterday it was special because the Honorable President of India was there with us. Today I'll be talking about the topic, that is evaluation of antiviral activity of Unani drugs against dengue virus. We are working on molecular biology of human viruses, and my fascination with viruses started in 2001 when I joined doctoral thesis at Department of Microbiology, All India Institute of Medical Sciences, where I started my thesis on respiratory syncytial virus. And since then, after completing my thesis, we are still working on human viruses including dengue, chikungunya, RSV, HIV, and lately SARS-CoV-2 also. Basically, our research group is studying three different aspects of these viruses. The first aspect is local and global evolutionary dynamics, in which we are trying to identify the viral strains from clinical samples, followed by different kind of phylogenetic Bayesian analysis. And this kind of work we have done for RSV, dengue, and chikungunya. The second aspect is we are trying to study different proteins of viruses, including the structural and non-structural proteins, and we are trying to characterize them by physical methods. And then in addition, we are also trying to identify the inhibitors against these viral proteins, and we are validating them by experimental and computational methods. We have expressed the G protein of RSV, HEV Y domain, NS3 protein of dengue virus, and N protein of SARS-CoV-2 virus. The third and very important aspect of our lab is we are trying to do the antiviral studies against different viruses of different drugs, and lately some Unani formulations. This particular project has been sanctioned to us by CCRUM, and in this particular project we are trying to evaluate the potential antiviral effect of different Unani drugs against dengue virus. We have been hearing about Unani formulations, plants-based extracts since yesterday, so I'll not talk much about this. Briefly about dengue fever, all of us know about dengue fever. It is a mosquito-borne viral infection which is transmitted by Aedes mosquito. It is prevalent in tropical and subtropical regions, and it is caused by dengue virus, which has structural and non-structural proteins. The structural proteins are part of the mature virion, and non-structural proteins helps in replication of the viruses. Although the vaccine dengue vaccine was approved by Sanofi Pasteur, was approved by FDA, but there are a lot of disadvantage of this virus. Until today, we do not have any drug against dengue virus. And this is the structure of the virus, and two main non-structural proteins, NS3 and NS5, which help in virus replication. We are targeting these two proteins. So what we have done in computational aspect of the study, we have selected NS3 and NS5 protein of dengue virus. We have taken their structure from PDB, and we have identified some ligands. These ligands are basically phytochemicals which are present in different Unani formulations. We have downloaded their structure, and then we have done virtual screening or docking of these ligands with the viral protein. Subsequently, we were able to identify some interesting ligands. Subsequently, we have to do some antiviral activity against these Unani formulations. So before we proceed to antiviral activity, we have to do the cytotoxic acid activity, which we have done by the standard MTT assay, and then we did the antiviral activity of the proposed drugs against the dengue virus. Now, we have taken a number of drugs for this particular project. I am just showing the result of one particular drug, that is Habe Papita. And in the computational studies, we have taken the structure of NS3 and NS5 protein, and we have taken around 100 phytochemicals from different databases, and they were docked with NS3 and NS5. In fact, we have done the docking with NS1, NS3, NS5, all three proteins. But here I am showing the data only for NS3 and NS5, and top 3 ligands were identified against both NS3 and NS5 protein of all the 4 different serotypes of dengue, that is dengue 1, 2, 3 and 4. And we have taken the ligands based upon their energy, that is between minus 7.1 to minus 10.7 kilocalories per mole, and we are doing MD simulations of some of these studies. Now this figure shows the results of the interaction between the ligand and the viral protein. The yellow colored ligand is interacting with the pink colored viral protein. This is for dengue 1 and this is also for dengue 1. The upper panel shows NS3 protein and the lower panel shows NS5 protein of dengue virus. Here you can see that the yellow colored ligand is having efficient interaction with the protein, and some of the interacting residues are mentioned here. Similarly, we have done the interaction between the NS3 protein and NS5 protein for dengue 2 virus. Then similarly, we have done for dengue 3 virus, and similarly we have done for the dengue 4 virus, and we have got some interesting results for these. For some of them we are going for MD simulation. Subsequently, we have done some cytotoxic studies for the Unani drugs. For this, we have taken the Vero cell lines and we have calculated the CC50 of these drugs. And then we went for the main experiment, that is the antiviral studies. For this, for doing antiviral studies, we have to culture the virus in mammalian cell lines. So we have taken the Vero cell lines and we maintained it by using standard protocol, and then we obtained 4 different serotypes of dengue 1, 2, 3 and 4 from NIV Pune. And these viruses were cultured in these cell lines using standard protocol, and they were confirmed by CPE and real-time PCR. For doing the antiviral studies, we have taken approximately the 80% of the confluent Vero cell line layer, and then we have infected these cell lines with the virus, and subsequently these plates were added with the drug. By particular concentration, we obtained this concentration from the cytotoxicity assay, and we incubated these infected plates for 5 to 7 days, and after 5 to 7 days we harvested the virus, and then we confirmed the virus by real-time PCR assays, and ribavirin was used as the control. We were able to see that addition of drug was able to inhibit the replication of viruses that range between approximately 10 to 25% of the total. So, yeah, last slide, conclusion. So, in conclusion, we have used Unani drugs and we were able to show that these Unani drugs, they are able to inhibit the viral replication. Now, these are the present and past students who are working in a research group. The work which I have showed was done by Aisha Tazeen. She'll be submitting her thesis, inshallah, next month. And these are our collaborators and our overseas collaborators. Funding, I'd like to acknowledge DBT, DST, CSR, UGC and CCRUM. This particular project is funded by CCRUM, and some part of the bioinformatic analysis we have done using CDAC Pune server. And thank you all of you for giving your kind attention.
Thank you so much, Professor Praveen, for that enlightening talk about the advances in virology with Unani medicine. I hand over now to Dr. Khan for some comments.
Thank you very much, Dr. Shama. One question asked to you, what is the use or the dose of Habbe Papita?
Actually, sir, we were suggested that we should use three different drugs for this particular
You
project.
mentioned only one, Habbe Papita, no?
I have not shown the data of other two. The other two are Khamira Marwarid, Habbe Papita and Sharbat Anjbar.
Compound drug, yeah, including individual, one by one?
No, compound.
Compound.
We are supposed to see the compound effect.
So dengue 1, 2, 3, you explain the dengue 1, 2 and 3.
1, 2, 3 and 4. Actually, these 4 are different viruses. So we have seen the inhibitory effect of these drugs on all the 4 viruses. There is slight difference. We consider them separate viruses. So we have done for all 4, for all the 3 drugs. But because of time limitation, I could not get the data for the other 2 drugs.
So thank you very much.
One more.
Any question asked?
One more
Anybody?
question.
Yes.
Madam, how you have chosen the concentration of that extract, Habbe Papita? Here.
Concentration?
Yes, madam.
We have used MTT assay to identify the concentration of drug which इसको ऑन कैसे करेंगे मेरा फोन है sorry about that.
After MTT-assay, you have found that
yes,
lower concentration.
From MTT-assay, we were able to identify what is the approximately concentration of the drug which is leading to 50% of the death of the cells. So what we have taken such concentration so that 80% of the cells are viable. Because to grow the cells, we need the viable cells. To grow the virus, we need viable cells. So we have taken approximately CC50 should be so that around 80% of the cells should be viable, because after virus infection, we have to incubate them for around 5 to 7 days for CPE.
Okay, madam. Ma'am, how you have chosen that extract? In which solvent you have made the extract?
We were provided these drugs by CCRUM.
Hydroethanolic or aqueous or methanolic?
Right now I don't remember uska kya tha, but I'm not able to recall that.
Thank you very much.
Okay, thank you.
Yeah, small. Can you come on the plant part actually? Plant, check third or fourth slide. So you have mentioned there this Phyllanthus neroi. Yeah, this one. Phyllanthus neroi is actually bhui amla. It's not amla. Amla is Umbilica officinalis.
Okay, so I'm sorry for that.
No,
I'll do this
it's
correction.
a suggestion only.
Yeah, I'll
because
do that correction.
identity is quite different morphologically and the molecular level. If you are using this amla, this is a hepatoprotective action. Nerori has hepatoprotective action, and amla, you all we know that it's a very.
I'm sorry for that. I will do this correction.
Yeah, so that's why identification and everything is very much important
Yes,
at the
I
initial
agree
stage
to
of
that.
the.
I agree to that.
Thank you.
Thank you all of you for your kind
much.
Thank you so much, Professor Parveen. I have the honor now to invite Professor Kumud Bala from the Department of Biotechnology, Amity University, Noida, and she will discuss the anti-cancerous efficacy of Unani formulation Majun Najah on cervical carcinoma.
Good afternoon. Good afternoon, everyone. I'm really honored to have this opportunity over here, and all the credit goes to Unani System of Medicine, from where we got the project, that is CCRUM. And I'm really thankful on behalf of my entire team that we have got this opportunity to do this work. So I would. No.
You want to change yourself by a pointer, or should you just command them? You have
Can I? Where? For this? Yeah, here.
Yeah, yes. You have to change.
Next. Back.
Sure.
Okay. Thank
you
you.
want to do it yourself, or
Yeah.
you can command them. They will also can change.
Okay. Next, if I'll see then.
They'll change from there.
Okay, I'll do it from here. Thank you so much. Thank you for the assistance. So, I'm from Amity University, that to be more precise, Amity Institute of Biotechnology. And I'm working, my specialization is cancer immunotherapy basically. And I have a team of 11 people in our lab. Beside the position of professor over there, I'm also deputy dean at Amity University. And I'll show you the entire group in the later slides. Let's start with what I have done and what I wish to show it here. My work is on the anti-cancerous efficacy of Unani formulation, that is Majunaja, on the cervical carcinoma. Basically, this medicine has been used for first of all, we will go for the cervical cancer. We all know that this is the fourth most prevalent cancer in the woman. And death, which is reported in 2020, was quite huge, that is 3,42,000. And this burden of this disease is increasing day by day. And the worst part is that this disease can be curable. However, because of the ignorance and lack of the medical facility to the rural people, this disease is spreading like anything. So what I want to say here, that the pathogen for this disease is HPV. We all know that HPV 16, HPV 18, they are the main culprit for this. Beside that, there are other strains also like HPV 45, 32 and all. So what I mean to say that because of the cancer burden and because of the side effect of the chemo drugs, we are looking for alternative medicine and also the herbal solution for that. That's why this study has been performed. Now, when we see the global burden also, though Asia has got the majority of the cases. However, when we go to India, 20.42% of the cervical cancers cases are there. And when we see the new cases, then you can also see that there are so many. I'm so sorry, I have not. When we go for the global scenario, then we see that the India is one of the most affected area, and I'm sorry, I think we are not getting coordinated now. This is the slide I was trying to focus on, and that rate is also quite high in India. Now coming to the next slide, we have got the Unani formulation, which is basically a semi-solid polyherbal Unani medicine, and it has been earlier used for hysteria, epilepsy, psychosis, schizophrenia, leprosy, mania and arthritis. However, we have been asked to see that whether this drug can be repurposed for treatment of the cervical carcinoma. So that's what we have generated the in vitro data for this purpose. Now, coming to the this formulation, we have 8 herbs in this formulation, which are listed over here in the proportion which is mentioned over here. Then we have got all these herbs collected from Khari Bawli. Then we have got the certification of these herbs done by the CSIR Institute, and then we have gone for this methodology, which is over here. We have collected the herbs, then we have gone for the sequential extraction of the herb, this formulation. So we have started from the N-hexane, then ethyl acetate, then methanol, and then ethanol. So this is the sequential extraction we have done. And then we have done the quantification of various phenolic content, flavonoid content, tannin content, And we have also checked the antioxidant capacity of various extract which we have got by the sequential extraction, along with the qualitative and quantitative, both we have done. Then we have done the separation on the TLC plate, and then we have gone for the characterization and the identification of the various phytoconstituent using GC mass chromatogram. And afterward, what we have done, we have taken the constituent which were present in the gas chromatography. On the basis of the gas chromatography, we have selected the ligands, and then we have taken all those receptors which get upregulated in cervical carcinoma, and then we have gone for the in silico docking to validate our result which we have found in our labs. So this is how we have progressed in this study. And this is the setup which we have got. We have gone for the sequential extraction. Then we have checked the percentage of yield in each extract. And we have got that the maximum yield was, we have observed in methanol. Then next we have gone for the component identification by various methods which are already listed and well known to all of us. And 4 extract we have taken: methanolic extract, ethanolic extract, ethyl acetate, and the N-hexane. Then the next we have gone for the TLC, where we have taken the standard drug also, like rutin, quercetin, curcumin, and piperine. For selection of these standard drug is on the basis of that 2 of them are the very strong antioxidant, and 2 of them are listed as an anti-cancerous drug. That's why these standard has been taken. So these standard has been used to check and validate the data which we have generated. Now coming to the DPPH assay which we have done for free radical scavenging capacity of that particular herb. So rutin and gallic acid has been taken. You can see the, I think this is the, yes. You can see here the proportion of the DPPH and the extract is 1:1, and this is the proportion which is showing the maximum amount of the antioxidant or the free radical scavenging activity. And then we have gone for the DPPH, quantification of the DPPF free radical scavenging, where we have found that methanol extract has the maximum capacity. Now coming to the next, we have done TPC, that is the total phenolic content estimation, where we have taken the gallic acid as standard, and this gallic acid has been used to compare the methanolic, ethanolic, and ethyl acetate and hexane extract quantification of the phenolic content. And we have found that the N-hexane has got the maximum phenolic content. Then we have done the total phenolic content, where quercetin has been taken as a standard, and we have found that N-hexane has shown the maximum concentration of it. Then we have gone for the tannin content, and we have taken gallic acid as our standard, and we have observed that ethyl acetate has got maximum concentration of the tannin. Then we have done the reducing power assay, where ascorbic acid has been taken as a standard, and the all extract has been estimated for their reducing power. Then we have gone for the ferric reducing assay, where ferrous sulfate has been used as a standard, and we have observed that our ethanolic extract has got the maximum activity. Now coming to the gas chromatogram, we have taken Majunaja N-hexane extract, and we have done the gas chromatographic analysis, and where the chromatogram has shown that these various components were present in it. So if you see that we have cyclopentanol 3-methyl, which was found in the maximum amount. Then we have beta U decimol, which was found. These 2 compound has been listed in various literature as a strongly antimicrobial agent and as a strong anti-cancerous agent. So these 2 compounds were present in the N-hexane fraction of Mazunaja. Now next, when we have gone for the ethyl acetate extract gas chromatogram, then we have observed that the silane, silane has got we have observed silane present, which is also again antimicrobial agent, and this we have D-sulfosinegrin, which was also present in a quantity which is mentioned over here. And these two are reported in various texts as antimicrobial agent and anti-cancer agent. So we do have in our non-polar fraction of Majunaja, the anti-cancerous compound and the strong antimicrobial compound. Then we have gone for the anti-cancerous efficacy on the HeLa cell line. Though there are other cell line also which we have like taken in consideration and evaluated the efficacy of it. However, I'm showing the data only related to the HeLa cell line. Here we have taken doxorubicin, the chemo drug, then the 5-fluorouracil also as a chemo drug, and then we have taken the antioxidant, which is quercetin and rutin, to see the efficacy of these extract. What we have observed that the ethyl acetate has shown very good result, and where the IC50 value have been shown here for the comparison. Now coming to next slide and the next result, where the cells has been shown over here. You can see the control cells, and the vehicle control also has been shown along with the doxorubicin result, 5-fluorouracils, along with the quercetin, rutin, and Majunaja ethanolic extract. You can see the value over here. So on the basis of the IC50 value, we can say that ethyl acetate is the best. Ethyl acetate fraction was the best for the anti-cancerous activity. Now coming to the next slide, which shows that what kind of cytotoxicity effect all these extract has got on the normal cell line. So we have taken the L929 cell, which is a normal fibroblast cell line, and we have evaluated its cytotoxicity effect, whether these extract have any cytotoxic effect on that or not. So here you can see that doxo and fluorouracil have shown major toxicity on these cells. However, ethyl acetate has all our fraction has no toxicity on L929. This is what we have observed. So it is very good for us that all these fraction of Majunaja has got anti-cancerous activity. However, they are not killing the normal cell line, which is L929. Now coming to next result, we have gone for the molecular. To validate our result, we have gone for the molecular docking. We want to see that the ligands which we have got on the basis of the gas chromatogram, are they actually binding to the receptor which get upregulated in case of the cervical carcinoma. So we have taken these receptors, that is the BCL2, NF-kappa beta, HER2, VEGF, then estrogen, progesterone, and glucocorticoid. And we have dogged all these ligand here. So you can see that the beta U desmol has got high binding energy with the receptor, that to HER2. And even silane has got highest binding energy over here with the HER2 receptor. So this is basically showing, yes, these drugs which are present in the gas chromatogram are actually anti-cancerous, and they are binding to the right receptor and inducing apoptosis in these cells. So this is very good.
2 more minutes.
Thank you, sir. I'll just finish up.
Okay.
So even the in silico docking has shown that we are actually going in the right direction, and Majunaja has got very good is a good consortium of antioxidant and anti-cancerous drug present in it. And now here I have shown various receptors and the ligand and their binding energy and where they are actually binding to. So what is the binding plane they are in? That's what I have tried to show here, and this is all about those binding planes. Now coming to the conclusion, we have found that on the basis of qualitative and quantitative analysis of various component phytoconstituent present in Majunaja, various fraction, which I have said that was N-hexane, ethyl acetate, methanol, and ethanol. We have seen that they are rich in flavonoid, alkaloid, tannin, as normally most of the herbal plants are. Now, the best part for us is that on the basis of GCMS analysis, we got to know that they have also got the anti-cancerous compound and very potent antimicrobial compound. So I can say that this formulation contains potent bioactive compounds with promising antioxidant and anti-cancer potential, particularly against cervical cancer. These findings suggest its potential application in the development of the novel therapeutic agent for cancer treatment. But however, I will also suggest that if in future we need to do further in-depth studies, including in vivo analysis and the molecular mechanism evaluation, so that we can say with full force that yes, Majun Naja can be repurposed for cervical carcinoma. Thank you all. This is all from my side. I'm open for question.
Thank you so much, Prof. Bala.
Anyone ask anybody question? Yes.
Madam, Majun Naja is basically used, correct me if I am wrong, for neurological disorders.
Yes, yes, yes. I have mentioned that epilepsy, hysteria, schizophrenia.
Anti-Savida. Otherwise, it is good means. I just want to know that when you are doing a cytotoxicity assay, how you are treating the cells. With extract, you are taking whole formulation, including sugar part.
No, 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 no. What we have done, we have excluded the sugar. This was what the CCROM has suggested us. So remove the sugar part. We have removed the sugar. That's why I have shown that in the same proportion, in which proportion the formulation is there, we have collected the herbs, we have certified it, then we have taken the same proportion, and then we have gone for...
Then somewhere, it's suggestion, then somewhere you have to mention that ingredients of Majun Naja apart from honey or sugar. Because it is making a big difference.
Yes.
We cannot call it Majun Naja anymore if we are not
Sir,
Thank you for the sorry for that. The instruction of the Director General. So please conclude the session.
Yeah.
So now go to the preliminary hall.
Yeah.
Thank you very much, all of you.
Everybody.
Sir, just 2 seconds. I would like to introduce my team also. Actually, I forgot, and I will be not pardoned before that in Mahila. Mohini Agrawal, she is SRF and CCRM project. She is here. She is sitting over there. And Manish is, can you say hi? And Manish Kumar, he is sitting over there. Manish, can you stand up and say hi? He is SRF and NMPV project, which we have got from NMPV. And all these students, Sarthak is here with us. Where are you, Sarthak? Yeah, here Sarthak is here, and there are one faculty is also there, and these are all lab trainees who are working on that. Thank you so much, sir. Thank you so much.
Just take it after some. Thank you so much, everybody. Thank you to all our distinguished chairpersons for conducting the scientific session so well and interactive, and all the eminent speakers for their insightful presentation. We are lagging behind time because we have to move towards the plenary hall for... can you please wait for the memento presentation? I was just continuing my talk. Please don't go. So now it's... Yes. Sure. Now I would like to, it's time to felicitate our eminent speakers with a memento. May I request Dr. Abdul Rauf, sir, to present a memento to Professor Farhan Jalil, sir. Can we have a big round of applause, please? They have given such a wonderful presentation. Can we have a big round of applause? Thank you. Thank you so much. I request Dr. Parvez Khan, sir, to present a memento to Dr. S.M. Raisuddin, sir. Can we have a big round of applause? I request Dr. Mujib Hussain, sir, to present a memento to Dr. Ahmad Ali, sir. Can we have a big round of applause? I request Dr. Parvez Khan sir to present a memento to Professor Shama Parveen ma'am. Can we have a big round of applause? Please, thoda sa taste सुनाई नहीं आ रहा, बिल्कुल. Thank you so much. I request Dr. Mujib Hussain sir to present a memento to Professor Kumud Bala ma'am. May I now request Dr. Parvez Khan to present a memento to our chair, Dr. Farukhrod Abdul Rauf sir. आप लोग सर परवेज सर आप देंगे. Can we have a big round of applause? They were the very good chairs in this scientific session. I request Mujib sir to present a memento to Dr. Parvez Khan sir. Can we have a big round of applause because he has asked so many questions and made the session so interactive. I request Dr. Bilal Ahmed sir to present a memento to Dr. Mujib Hussain sir. The enthusiastic one. Can we have a big round of applause? With this, we are towards the end. Thank you so much for coming to this session. And I would request, before moving, I would like to request our speakers to please have a photograph with the chairs. I would request everybody to please proceed towards the plenary hall for the valedictory session, as our honored guests are here already in the plenary hall. Kindly please proceed towards the